Welcome. Friends, I'm so glad you're here. Whether you're drifting towards sleep, unwinding after a long day, or simply looking for a quiet companion, you've found the right place. Before we begin, I'd love to know where in the world are you listening from. Perhaps you're wrapped in a blanket, sitting by a softly lit window, or resting somewhere under an open sky. Wherever you are, I hope you're warm, comfortable, and ready to wander with me into Earth's deepest past to meet the inhabitants who lived here long before humans ever existed. If you find peace or curiosity in these gentle stories, please consider liking the video, subscribing to the channel, or sharing it with someone who might need a moment of stillness too. And to those who've already joined along, thank you. 4.5 billion years ago, Earth breathes through volcanic pores scattered across its molten surface. Steam rises from countless fissures, carrying with it the elements that will someday become bone and leaf and thought. The atmosphere thickens slowly, molecule by molecule, as gases escape from the planet's interior like sighs from some vast, dreaming creature. Water vapor, methane, ammonia, and hydrogen sulfide, a cocktail that would poison any modern lung drift through skies lit only by lightning and the dull red glow of cooling lava. The young planet spins beneath a smaller, brighter sun, its days lasting only 5 hours as it rotates with the frantic energy of youth. The moon hangs 3 times closer than it will in humanity's distant future, pulling tides that surge across continents with titanic force. These enormous tides stretch for hundreds of miles inland, creating vast tidal flats where seawater mixes with mineral-rich runoff from the cooling continents. Here, in these rhythmic pools that fill and empty twice each brief day, chemistry begins its first experiments with complexity. Comets and asteroids still rain down regularly, their impacts adding both destruction and the precious gift of water. Each collision melts rock into glass, but also delivers the organic compounds that hitchhiked through space for millions of years. Carbon, that most sociable of atoms, begins its long conversation with hydrogen, oxygen, and nitrogen in puddles that form and boil away with each impact. The late heavy bombardment brings not just destruction, but raw materials, amino acids frozen in ice, simple sugars locked in meteorite hearts, nucleotide bases that rode the stellar wind across impossible distances. In laboratory experiments, scientists recreate these ancient conditions with electric sparks jumping through carefully mixed gases, watching as amino acids condense like dew on glass walls. The Miller-Urey experiment, repeated countless times since the 1950s, shows how readily life's building blocks form when energy meets the right atmospheric mixture. But the real Earth's atmosphere remains a puzzle. Was it truly reducing, rich in hydrogen and methane? Or had oxygen already begun to seep from water molecules split by ultraviolet light? The uncertainty hangs like morning mist over our understanding of life's origins. Scientists call these earliest organic molecules the products of prebiotic chemistry, though the boundary between chemistry and biology remains beautifully unclear. Simple sugars crystallize in cooling pools, their 6-membered rings forming spontaneously when formaldehyde concentrations reach critical thresholds. Amino acids link hands in short chains before heat breaks them apart again. Each temporary peptide, a brief sentence in chemistry's expanding vocabulary. Nucleotides, the building blocks of genetic code, assemble spontaneously when conditions align just so. Their phosphate backbones forming chains that will one day carry the instructions for every living thing. But these molecules face constant dissolution. Each rainfall dilutes their carefully concentrated solutions. Each dry spell crystallizes them into useless salts. Each temperature spike breaks their delicate bonds, sending complex structures tumbling back into simpler components. The early Earth operates like a vast laboratory with no patient experimenters, where every promising reaction gets interrupted by the next geological catastrophe. Volcanic eruptions vaporize entire inland seas. Asteroid impacts melt continents back to slag. The planet's surface churns and reforms like a pot of slowly boiling soup. The irony settles like sediment. Life's ingredients are everywhere, scattered across a world too violent to sustain life itself. Every puddle becomes a brief experiment in molecular complexity before evaporating into the thin air. Every tide pool assembles and disassembles the same fundamental components, like a patient craftsman learning the same skill over and over again. Only took a few 100 million years of practice. Only required turning every drop of water on the planet's surface into a temporary laboratory where molecules learn to dance together in increasingly complex patterns. Yet, even in this chaos, patterns emerge. Organic molecules show preferences, forming more readily in some environments than others. Clay minerals act as gentle catalysts, their layered surfaces providing templates where larger molecules can assemble with less energy required. Montmorillonite clays, in particular, seem to encourage RNA-like polymers to form longer chains, their negatively charged surfaces concentrating phosphate-rich molecules like tiny magnetic fields gathering iron filings. Lightning provides the dramatic sparks, but the real work happens in the quiet spaces between storms, in the shallow margins where water meets rock meets air, where chemistry begins its first tentative steps towards something more organized. Hydrothermal vents begin appearing along the ocean floor as the first stable oceanic crust forms. These underwater geysers pump superheated water rich in dissolved metals and sulfur compounds into the cold depths. Around their mouths, temperature gradients create natural reaction chambers where complex chemistry can proceed without the constant interruption of surface bombardments. Iron-sulfur minerals precipitate in delicate towers, their surfaces catalyzing reactions that mirror some of the fundamental metabolic pathways still used by life today. The deep ocean becomes Earth's first stable laboratory, protected from cosmic violence by miles of water. In these protected depths, concentration becomes possible for the first time. Organic molecules no longer face immediate dilution or destruction. They can accumulate, interact, form larger assemblies that persist long enough for evolution's first stumbling experiments with self-organization. Lipid-like molecules begin forming spontaneous vesicles, soap bubbles essentially, that create the first boundaries between inside and outside, self and not self. These protocells lack the sophisticated machinery of true life, but represent the crucial conceptual leap from scattered molecules to organized systems. The mystery that haunts this earliest chapter remains unsolved. How did non-living matter first organize itself into patterns complex enough to be called alive? The gap between the most sophisticated organic molecule and the simplest living cell still yawns wide in the fossil record, a quiet riddle that 4 billion years of evolution have not fully answered. No known chemical reaction can bridge this chasm in a single step, yet somehow it was crossed, leaving no trace of the intermediate stages that must have existed. Perhaps life emerged not once, but countless times, with only the most successful experiments surviving to leave descendants. Perhaps the transition happened so gradually that no clear boundary exists between living and non-living systems, only degrees of organization, complexity, and self-maintenance that shade imperceptibly into what we call life. The young Earth keeps its secrets buried beneath billions of years of subsequent geology, leaving us to wonder at this first great mystery of existence, how chemistry learned to dream. And in dreaming, became something more than chemistry alone. The first stirring of chemistry happens not in a single moment, but across millions of years of patient accumulation. In shallow seas that taste of iron and sulfur, carbon-based molecules begin their slow dance toward complexity. The oceans have grown deeper now, fed by countless comets melting into steam and rain. These ancient waters hold 10 times more dissolved iron than any modern sea, giving them a rusty green tint that would look alien beneath today's blue skies. The dissolved metals act as catalysts, speeding reactions that might otherwise take geological ages to complete. Simple organic compounds drift through these primordial waters like microscopic seeds waiting for the right conditions to sprout. Formic acid, the simplest organic acid, forms readily wherever carbon monoxide meets water vapor over hot volcanic surfaces. Formaldehyde molecules link together spontaneously when concentrations reach critical thresholds, building longer and longer carbon chains like a molecular conga line that never quite decides where to stop dancing. Hydrogen cyanide, deadly poison to future life forms, serves as a crucial building block in this oxygen-free world, readily combining with other molecules to form amino acids and nucleotide bases. In the laboratory, biochemists have recreated these ancient seas in glass vessels, watching as simple molecules undergo what they call combinatorial chemistry, every possible reaction happening simultaneously in the same solution. The results surprise even experienced researchers given enough time and the right conditions. Organic chemistry displays an almost magnetic attraction toward the kinds of molecules that life will eventually require. Ribose sugars form preferentially over other possible configurations. Amino acids show a bias toward the 20 varieties that proteins will later adopt. Nucleotide bases assemble with surprising frequency, as if chemistry itself holds some memory of life's future needs. But these early seas face a fundamental problem that chemists call the dilution dilemma. Even when organic molecules form successfully, they immediately disperse throughout the vast ocean volume, making further reactions statistically unlikely. It's like trying to have a conversation in a library where every word gets whispered into a different room. The components of life's first sentences scatter before they can find each other to form meaningful phrases. The mathematics of molecular encounters suggests that complex chemistry should be nearly impossible in such dilute solutions. Yet the young Earth provides solutions that no laboratory can fully replicate. Evaporating tide pools concentrate dissolved molecules 1,000-fold, creating natural test tubes where chemistry can proceed at reasonable rates. Mineral surfaces act as gathering places where specific types of molecules accumulate, like molecular coffee shops where certain regulars always meet. Clay particles, in particular, develop an uncanny ability to attract and organize organic molecules on their surfaces, their crystalline structures providing templates that encourage longer polymers to form. Pyrite formation becomes one of the planet's most important chemical engines during this period, when iron-rich water meets hydrogen sulfide around hydrothermal vents. Iron sulfide crystals precipitate with tremendous energy release. This energy doesn't dissipate as waste heat, but drives the formation of increasingly complex organic molecules in the surrounding water. The reaction produces the same kinds of carbon compounds that living cells will later use in their most fundamental metabolic pathways, suggesting that life's basic chemistry might have been forged in the furnaces of mineral formation. The irony of abundance appears. Earth's early oceans contain all the ingredients for life, but in such vast volumes that meaningful interactions remain vanishingly rare. Every promising chemical reaction gets immediately diluted by the surrounding seawater. Every complex molecule that forms faces a lottery of destruction versus further complexity. With the odds heavily stacked against advancement, only took chemistry several 100 million years to solve this particular puzzle, only required the invention of concentration mechanisms that could gather scattered molecules into productive communities. Lipid molecules begin appearing with increasing frequency in fossil chemical signatures from this period. These simple, fatty compounds, ancestors of the membranes that will eventually surround every living cell, form spontaneously when long-chain organic acids encounter the right pH conditions. In laboratory demonstrations, lipids assemble themselves into hollow spheres called vesicles without any external guidance, their hydrophobic tails clustering together while their water-loving heads face outward toward the surrounding solution. These protocells can grow, divide, and even compete for resources in ways that foreshadow biological evolution. Temperature cycling emerges as another crucial factor in early molecular evolution. Day-night temperature swings, caused by the planet's rapid rotation, create natural thermal engines that drive chemical reactions forward during hot periods and allow products to stabilize during cool phases. This cycling prevents the chemical equilibrium that would otherwise halt all molecular progress, forcing reactions to proceed in one direction rather than simply reversing themselves endlessly. The young Earth's thermal instability becomes chemistry's first evolutionary pressure. RNA-like molecules begin appearing in the fossil biochemical record, though whether these represent true RNA or simply RNA-like polymers remains beautifully uncertain. These early nucleic acids face immediate challenges. They degrade rapidly in water, especially at the high temperatures common on the early Earth. Yet they also display remarkable self-organizing properties, with certain sequences showing the ability to catalyze their own formation from available building blocks. This autocatalytic behavior represents chemistry's first tentative steps towards self-replication. The transition metals dissolved in ancient seawater play increasingly important catalytic roles as molecular complexity grows. Iron, nickel, cobalt, and molybdenum atoms begin incorporating themselves into organic frameworks, creating primitive metalloproteins that can facilitate reactions impossible with organic chemistry alone. These early metal-organic complexes will eventually become the active sites of enzymes, but for now they exist as simple catalysts that help the first complex molecules maintain their integrity long enough to participate in further reactions. Deep ocean chemistry proceeds along different pathways than surface reactions, protected from ultraviolet radiation and temperature extremes, but also isolated from many energy sources. Here, in the permanent darkness of the abyss, chemosynthetic pathways begin emerging reactions that extract energy from chemical gradients rather than light. These depth-dwelling chemical processes will eventually give rise to entire ecosystems that operate independently of solar energy. Communities of organisms that live by consuming the planet's own chemical reserves. The mystery that defines this period centers on selectivity. Why did early Earth's chemistry favor certain molecular configurations over others? Random reactions should produce random results. Yet the fossil chemical record shows clear preferences for the specific types of molecules that life would later adopt. Some unknown selection pressure seems to guide molecular evolution toward biologically relevant compounds billions of years before biology itself appears. Whether this represents fundamental chemical constraints or evidence of earlier, simpler forms of life remains one of paleochemistry's most intriguing puzzles. In warm tidal pools that taste of salt and metal, the first molecules learn to make copies of themselves. RNA emerges from the chemical soup not as a single dramatic event, but as a gradual coalescence of possibilities that have been building for hundreds of millions of years. These early ribonucleic acids stretch like molecular sentences across the curved surfaces of clay particles, their four-letter alphabet spelling out instructions that chemistry itself seems eager to follow. Unlike the proteins that will later dominate cellular machinery, RNA performs a peculiar double duty. It stores information like a library and catalyzes reactions like a factory, becoming both the blueprint and the craftsman of early molecular life. The discovery of ribozymes in modern organisms provides a window into this ancient RNA world. These catalytic RNA molecules can cut, splice, and synthesize other RNA strands with the precision of molecular scissors and glue. In laboratory experiments, scientists have evolved artificial ribozymes that can replicate themselves almost indefinitely, given a steady supply of nucleotide building blocks. The implications ripple backward through deep time if RNA can function as both gene and enzyme in carefully controlled laboratory conditions. Perhaps it once dominated an entire planetary ecosystem where proteins and DNA had not yet been invented. But RNA faces formidable challenges in the harsh environment of early Earth. The molecule degrades rapidly in hot water, its phosphate backbone hydrolyzing within hours at temperatures that were commonplace on the young planet. Magnesium ions, abundant in ancient seawater, can stabilize RNA structures, but only within narrow concentration ranges too little and the molecules fall apart. Too much, and they precipitate out of solution entirely. The early RNA world must have existed in a delicate balance between destruction and preservation, with successful replicators emerging only in the rare microenvironments where conditions aligned perfectly. Hydrothermal vents begin playing an increasingly important role as RNA incubators during this period. The temperature gradients around these underwater geysers create natural thermocycles that can drive RNA synthesis during hot phases and allow the products to fold into stable structures during cooler periods. Modern experiments using similar heating and cooling cycles can evolve simple RNA replicators within a few dozen generations, suggesting that the early Earth's thermal instability might have been evolution's first engine rather than its enemy. Ice becomes an unexpected ally in RNA's struggle for stability. When shallow pools freeze during the planet's occasional cold snaps, the growing ice crystals exclude salts and other dissolved minerals, creating pockets of pure water where delicate RNA molecules can survive much longer than usual. These frozen sanctuaries preserve molecular experiments through periods of environmental chaos, allowing evolution to resume when conditions improve. The irony of ice preserving life's first attempts at self-replication adds a poetic twist to an already unlikely story. The four bases that make up RNA adenine, guanine, cytosine, and uracil each form through different chemical pathways. Yet they appear together in the fossil record with surprising regularity. Adenine emerges readily from hydrogen cyanide reactions, while guanine requires more complex synthetic routes involving multiple steps and intermediate compounds. Cytosine and uracil follow their own unique formation pathways. Creating a puzzle for origin of life researchers, why did early chemistry consistently produce these four specific bases rather than the dozens of other possible nucleotide variants? Laboratory studies reveal that these four bases possess unique properties that make them ideal for information storage and transfer. They form the strongest and most specific hydrogen bonds with their complementary partners, creating the molecular recognition that allows accurate copying during replication. Other possible bases either bond too weakly to ensure faithful copying or too strongly to allow the strand separation required for replication. Chemistry seems to have discovered the optimal solution to the information storage problem through blind trial and error across geological timescales. RNA's ability to fold into complex three-dimensional shapes gives it catalytic powers that simple linear molecules lack. Ribozymes adopt intricate structures with active sites that can bind specific substrates and accelerate chemical reactions by factors of millions. The most remarkable ribozymes can synthesize other RNA molecules from scratch, essentially functioning as molecular Xerox machines that copy genetic information without requiring protein enzymes. These ancient molecular tools represent chemistry's first successful attempt at creating self-sustaining systems. The concentration problem that plagued earlier chemical evolution finds partial solutions in the RNA world. Long RNA molecules can bind to clay mineral surfaces through their negatively charged phosphate groups, creating local concentrations that are thousands of times higher than the surrounding solution. These surface-bound RNA communities can exchange sequences through horizontal transfer, sharing successful innovations across the entire population rather than limiting them to direct descendants. Competition emerges as different RNA sequences vie for limited nucleotide resources in their shared environment. Faster replicators outcompete slower ones, driving the evolution of increasingly efficient copying mechanisms. Error rates during replication introduce variations that natural selection can act upon, creating the first crude evolutionary algorithm running on molecular hardware. The most successful RNA replicators are not necessarily the most accurate, but rather those that strike the optimal balance between fidelity and speed. Parasitic RNA sequences begin appearing as inevitable byproducts of this molecular competition. These shorter sequences replicate faster than their hosts by borrowing copying machinery while contributing nothing in return. The evolutionary arms race between hosts and parasites drives increasing complexity in RNA systems, with hosts developing mechanisms to exclude parasites and parasites evolving new strategies to evade detection. This molecular conflict will later become one of the major drivers of cellular complexity. The mystery that haunts the RNA world concerns its relationship to the DNA protein systems that will eventually replace it. Modern cells still retain ribozymes in crucial positions. The ribosome itself is fundamentally a ribozyme that synthesizes proteins, suggesting that the transition from RNA to DNA protein never completely occurred. Perhaps the RNA world didn't end, but rather got incorporated into larger, more complex systems, its molecular fossils still visible in every living cell. The question of how and why this transition occurred remains one of evolution's most intriguing puzzles. A molecular detective story written in the language of nucleotide sequences. Only took a few 100 million years for chemistry to invent information, storage, catalysis, and self-replication all in the same molecular package. Only required countless failed experiments scattered across the ocean floor before stumbling upon the right combination of stability and reactivity. Only needed time measured in geological epochs for molecules to learn the first lessons of evolution, copy yourself accurately enough to maintain identity, but not so accurately that change becomes impossible. Membranes form like whispered secrets along the boundaries between water and oil, marking the first true distinction between self and not self in Earth's history. Lipid molecules, those dual-natured compounds with water-loving heads and water-fearing tails, begin congregating in the warm shallows where organic chemistry has been experimenting with increasingly sophisticated arrangements. These amphiphilic molecules solve an ancient puzzle that chemistry alone could never resolve: how to create stable compartments that can concentrate valuable resources while remaining permeable enough to allow continued growth and exchange with the outside world. The spontaneous formation of lipid bilayers represents one of physics' most elegant solutions to the problem of molecular organization. When fatty acids reach critical concentrations in ancient seawater, they self-assemble into hollow spheres without requiring any external template or guidance. The hydrophobic tails cluster together in the membrane interior, excluding water molecules and creating a barrier that can maintain chemical gradients across nanometer scale distances. The hydrophilic heads face outward, interfacing peacefully with the aqueous environment while maintaining the membrane's structural integrity. These first protocells emerge as microscopic soap bubbles filled with concentrated solutions of RNA, amino acids, and other organic molecules that would otherwise disperse throughout the vast ocean. Laboratory experiments demonstrate that such vesicles can grow by incorporating additional lipids from their surroundings and can divide spontaneously when mechanical forces stretch them beyond stable configurations. The division process randomly distributes internal contents between daughter vesicles, creating the first crude mechanism for hereditary transmission of chemical compositions across generations. Fatty acid membranes display remarkable tolerance for the harsh conditions of early Earth. Unlike the complex phospholipids that stabilize modern cell membranes, simple fatty acids can maintain bilayer structures across wide temperature ranges and pH conditions. They self-repair when damaged by radiation or chemical attack, automatically sealing small tears through the same hydrophobic forces that created them initially. This robustness proves crucial during an era when environmental conditions fluctuate wildly and protective mechanisms remain primitive. The concentration effect inside protocells transforms the dynamics of molecular evolution dramatically. Chemical reactions that would require millions of years in dilute ocean water can proceed in minutes or hours when reactants are confined within membrane-bounded spaces only micrometers in diameter. RNA replication rates increase by orders of magnitude, allowing evolutionary processes to accelerate from geological to biological timescales. The first cells essentially function as natural test tubes that permit rapid chemical experimentation under controlled conditions. Selective permeability emerges as membranes begin discriminating between different types of molecules attempting to cross their boundaries. Small, uncharged molecules like water and dissolved gases pass through freely, while larger or charged compounds face varying degrees of resistance. This selective filtering allows protocells to maintain internal chemical compositions that differ significantly from their external environment, creating the first primitive metabolisms based on controlled chemical gradients rather than random molecular encounters. The development of transmembrane gradients provides protocells with their first reliable energy source independent of external chemical reactions. Concentration differences across the membrane store potential energy that can drive otherwise unfavorable reactions, much like water behind a dam waiting to turn turbines. Early cells learn to harness these gradients through primitive transport proteins that couple energetically favorable processes to unfavorable ones, creating the first molecular machines that convert one form of energy into another. Competition between protocells introduces evolutionary pressures that favor increasingly sophisticated membrane compositions. Vesicles with more stable membranes persist longer in challenging environments, while those with more permeable boundaries can acquire resources more rapidly during favorable conditions. Natural selection begins operating at the cellular level for the first time, rewarding innovations in membrane chemistry, internal organization, and resource acquisition strategies. The ironies multiply like cellular divisions. The same forces that create cellular boundaries also threaten to trap protocells in static configurations that resist further evolution. Membranes that become too impermeable cannot acquire new resources or eliminate waste products effectively. Those that remain too leaky lose their carefully accumulated internal chemistry to the surrounding environment. Only took evolution several 100 million years to discover the optimal balance between isolation and exchange that allows continued growth while maintaining cellular identity. RNA begins playing increasingly important roles in membrane function as protocells develop more sophisticated boundary control mechanisms. Certain RNA sequences show affinity for lipid membranes, potentially serving as primitive membrane proteins that facilitate selective transport across cellular boundaries. The interaction between nucleic acids and lipids creates feedback loops where membrane composition influences internal RNA evolution. While RNA innovations modify membrane properties in return, size limitations become apparent as protocells attempt to grow beyond optimal dimensions. Simple diffusion can only transport materials across cellular interiors efficiently when distances remain small, limiting early cells to diameters measured in micrometers. Larger protocells face internal transport bottlenecks that starve their central regions of essential resources, creating evolutionary pressure for innovations in intracellular organization and transport that will eventually lead to complex cellular architectures. The fossil record preserves indirect evidence of these earliest cellular experiments in the form of isotopic signatures that suggest biological carbon fixation processes operating within membrane-bounded systems. Carbon-12 becomes preferentially incorporated into organic compounds through enzymatic pathways, leaving isotopic fingerprints that persist in ancient rock formations. These chemical fossils provide tantalizing glimpses of cellular metabolism operating billions of years before the first recognizable cellular structures appear in the geological record. Temperature cycling continues playing crucial roles in protocell evolution, with thermal expansion and contraction creating mechanical stresses that drive cellular division processes. The early Earth's rapid day-night temperature swings essentially function as a planetary heartbeat that pumps protocells through repeated cycles of growth and reproduction. This environmental forcing mechanism ensures that successful cellular innovations spread rapidly through populations while failed experiments are quickly eliminated through thermal stress. The mystery of homochirality begins manifesting as protocells develop increasingly sophisticated internal chemistry. Modern life exclusively uses left-handed amino acids and right-handed sugars, but the chemical reactions that produce these compounds in laboratory settings create equal mixtures of both mirror image forms. Somehow, the transition from chemistry to biology involved a symmetry-breaking event that selected one-handedness over the other, though the mechanism responsible for this selection remains hidden in the deep past. Perhaps the most profound innovation of the protocellular era lies not in any specific molecular mechanism, but in the fundamental concept of individuality itself. Before membranes, chemical evolution operated as a collective enterprise where successful innovations could spread freely throughout the global ocean. The advent of cellular boundaries created the first true biological individuals, discrete entities that could evolve independently while competing for shared resources. This transition from communal chemistry to individual biology represents one of the most significant conceptual leaps in the history of life, establishing the basic framework within which all subsequent evolution would operate. Prokaryotes emerge as Earth's first true citizens, microscopic pioneers that colonize every available habitat with the patience of geological time and the persistence of molecular machinery perfected over hundreds of millions of years. These simple cells, lacking the membrane-bound nuclei that will define their more complex descendants, spread through ancient oceans like living dust, each individual carrying the accumulated wisdom of countless generations encoded in circular chromosomes that float freely within their cytoplasm. Their success lies not in complexity, but in elegant simplicity, streamlined cellular designs that can function reliably across temperature ranges that would destroy more sophisticated organisms. Bacteria and archaea diverge along separate evolutionary pathways during this ancient period, though both groups maintain the fundamental prokaryotic architecture that proves so remarkably successful. The differences between them remain subtle, but profound bacteria develop cell walls made of peptidoglycan, a mesh-like polymer that provides structural support while remaining flexible enough to accommodate growth and division. Archaea construct their boundaries from pseudopeptidoglycan and other unique compounds, creating cellular walls that can withstand extreme conditions that would dissolve bacterial membranes within minutes. The metabolic diversity that prokaryotes achieve during their early proliferation staggers modern researchers attempting to catalog the biochemical innovations of this period. Some bacteria learn to extract energy from sulfur compounds bubbling up from hydrothermal vents, creating the first chemosynthetic ecosystems that operate independently of solar radiation. Others develop the ability to reduce iron oxides dissolved in seawater, essentially breathing rust and leaving behind distinctive banded iron formations that serve as geological markers of early biological activity. Still others discover how to fix atmospheric nitrogen into ammonia, making this essential element available to all subsequent life forms. Hyperthermophiles establish themselves around volcanic hot springs and underwater vents where temperatures regularly exceed the boiling point of water at sea level. These extreme organisms develop heat shock proteins and other molecular mechanisms that maintain cellular function at temperatures that would instantly denature the proteins of more conventional life forms. Their existence pushes the boundaries of what scientists consider compatible with life, suggesting that the habitable zone around early Earth extended into environments previously thought sterile. Acidophiles colonize pools of sulfuric acid where pH levels drop below those found in car batteries. Their cellular machinery adapted to function in conditions that would dissolve most metals. These organisms maintain internal pH levels near neutrality through sophisticated buffering systems and specialized membrane pumps that actively exclude excess hydrogen ions. Their success in such hostile environments demonstrates the remarkable plasticity of cellular biochemistry when shaped by extreme selective pressures over evolutionary time. Psychrophiles adapt to the opposite extreme, developing antifreeze proteins and modified membrane compositions that remain functional at temperatures approaching the freezing point of seawater. These cold-adapted organisms slow their metabolic rates to match the reduced kinetic energy available at low temperatures, essentially entering a state of controlled hibernation that can persist for thousands of years when conditions become unfavorable. Their strategies for cold survival will later prove crucial during Earth's periodic ice ages. The irony of invisible conquest becomes apparent. The most successful organisms in Earth's history remain largely unnoticed by casual observation, their presence detectable only through sophisticated chemical analysis and microscopic examination. Prokaryotes achieve global domination not through size or strength, but through sheer numbers and metabolic versatility. A single gram of soil contains billions of bacterial cells representing dozens of different species, each pursuing its own ecological strategy within the microscopic spaces between mineral particles. Horizontal gene transfer emerges as one of prokaryotes' most powerful evolutionary tools, allowing beneficial mutations to spread rapidly through entire populations regardless of traditional reproductive barriers. Bacteria exchange genetic material through transformation, taking up DNA fragments from their environment and incorporating useful sequences into their own genomes. Conjugation allows direct transfer of genetic material between living cells through temporary cellular bridges, while transduction uses viruses as vectors to carry genes between distantly related organisms. Biofilm formation becomes a crucial survival strategy as prokaryotic communities learn to cooperate in ways that foreshadow the multicellular organization of later life forms. Individual bacteria secrete extracellular polymers that bind cells together into cohesive mats with emergent properties exceeding those of isolated individuals. These bacterial cities can defend against environmental stresses, facilitate nutrient sharing, and coordinate collective responses to changing conditions through chemical signaling systems that operate like primitive nervous systems. Stromatolites begin forming in shallow marine environments as cyanobacteria trap and bind sediment particles within their growing biofilms. These layered structures represent some of the earliest visible evidence of biological activity preserved in the fossil record. Their characteristic dome shapes and laminated internal architecture providing unmistakable signatures of ancient microbial communities. Modern stromatolites still form in a few isolated locations, offering living windows into bacterial ecosystems that dominated Earth for billions of years. Endospore formation allows certain bacterial lineages to survive environmental catastrophes that would otherwise drive them to extinction. These dormant cellular forms can withstand radiation levels that would kill most organisms instantly, temperature extremes that span hundreds of degrees, and chemical conditions that would dissolve unprotected cellular machinery. Endospores can remain viable for millions of years, essentially allowing organisms to skip forward through geological time until favorable conditions return. The development of chemotaxis gives prokaryotes their first primitive sensory capabilities, allowing them to detect chemical gradients and move toward favorable conditions or away from harmful substances. Bacterial flagella rotate like molecular propellers, driven by proton gradients across the cell membrane that function like biological batteries. The coordination between sensory detection and motor response represents one of the earliest examples of integrated biological behavior, primitive versions of the stimulus response patterns that will later evolve into complex nervous systems. Quorum sensing emerges as bacterial populations reach critical densities that make collective action advantageous. Individual cells produce and detect signaling molecules whose concentrations reflect local population density, triggering coordinated responses when bacterial communities reach sufficient size. This chemical communication system allows prokaryotic populations to behave like multicellular organisms when circumstances favor collective action while maintaining the advantages of unicellular independence during normal conditions. Only took prokaryotes a few billion years to explore virtually every possible biochemical niche on the planet. Only required endless experimentation with cellular architecture and metabolic pathways to discover solutions for survival under conditions ranging from near absolute zero to several hundred degrees above the boiling point of water. Only needed the patient accumulation of countless small innovations to transform a sterile planet into a living world capable of supporting the complex ecosystems that would eventually follow. The mystery that pervades prokaryotic evolution centers on the transition from chemical systems to truly biological ones. Despite their apparent simplicity, even the most primitive bacteria possess molecular machinery of staggering complexity: ribosomes, containing thousands of precisely arranged atoms, metabolic pathways involving dozens of specialized enzymes, and genetic regulatory systems that rival modern computers in their sophistication. How this complexity emerged from the simpler chemical systems that preceded it remains one of evolution's most challenging puzzles, a gap in our understanding that spans the crucial transition from chemistry to biology itself. Cyanobacteria begin their quiet revolution in the shallow seas, their green-blue filaments weaving through ancient stromatolites like living thread through geological tapestry. These unassuming organisms possess a molecular innovation that will transform the planet more profoundly than any volcanic eruption or asteroid impact the ability to split water molecules using sunlight, releasing oxygen as a waste product in a process that modern science calls oxygenic photosynthesis. For the first time in Earth's history, living creatures harness the inexhaustible energy of stellar radiation while simultaneously producing the gas that will eventually become essential for complex life. The biochemical machinery that enables this process staggers in its sophistication. Photosystem 2, the protein complex responsible for water splitting, contains over 20 different subunits arranged with atomic precision around clusters of chlorophyll molecules and metal cofactors. When photons strike the reaction center, electrons get excited to higher energy levels, creating the electrical potential needed to tear apart water molecules against their natural chemical stability. The liberated oxygen atoms combine to form molecular oxygen gas, while the remaining hydrogen atoms provide the reducing power needed to convert carbon dioxide into organic compounds. Cyanobacterial mats spread across the continental shelves like vast underwater prairies, their photosynthetic activity visible from space as they release streams of oxygen bubbles into the overlying water column. These early photosynthetic communities achieve productivity levels comparable to modern forests. Fixing carbon dioxide at rates that begin measurably altering atmospheric composition for the first time in Earth's history. The oxygen they produce initially dissolves in seawater, where it encounters vast reserves of dissolved iron that have accumulated over billions of years. The reaction between oxygen and iron creates one of the most spectacular geological events in Earth's history, the precipitation of banded iron formations that will become some of the planet's most distinctive rock layers. As oxygen concentrations reach saturation levels in ancient oceans, iron oxides begin crystallizing out of solution in massive quantities, creating alternating bands of red and black minerals that record the rhythm of early photosynthetic activity. These formations represent the largest iron ore deposits on Earth, their distinctive banded patterns serving as both economic resources and geological calendars of early biological productivity. For most existing life forms, the rise of atmospheric oxygen represents an unprecedented catastrophe. Oxygen, despite its future importance for complex life, behaves as a potent cellular poison for organisms adapted to the reducing conditions that have dominated Earth's early history. The gas readily forms toxic free radicals that damage cellular membranes, denature proteins, and destroy nucleic acids faster than most organisms can repair the damage. Anaerobic bacteria that have thrived for billions of years find themselves facing chemical warfare from their own atmosphere. The Great Oxygenation Event unfolds over several 100 million years, but its effects prove irreversible once atmospheric oxygen concentrations reach critical thresholds. Methane, a greenhouse gas that has helped maintain warm global temperatures despite the fainter early sun, reacts with oxygen to form carbon dioxide and water vapor. This atmospheric chemistry change reduces the greenhouse effect, triggering a global cooling event that some researchers believe led to Earth's first major ice age. The irony compounds the first organisms to tap solar energy directly inadvertently plunge the planet into its first global winter. Mass extinction strikes the anaerobic communities that have dominated Earth's biosphere since life's earliest appearances. Entire lineages of bacteria and archaea disappear from the fossil record as oxygen concentrations climb toward modern levels. The organisms that survive do so either by retreating to oxygen-free refugia in deep sediments and isolated water bodies, or by evolving biochemical defenses against oxygen toxicity. The enzyme catalase, which breaks down hydrogen peroxide before it can damage cellular components, becomes essential equipment for life in an oxygenated world. Aerobic respiration emerges as some organisms learn to harness oxygen's destructive power for their own metabolic purposes. Instead of fighting oxygen's tendency to steal electrons from organic molecules, These innovators channel the process into controlled biochemical pathways that extract far more energy from nutrients than anaerobic metabolism can provide. The complete oxidation of glucose through aerobic respiration yields 38 molecules of ATP per sugar molecule, compared to only 2 from anaerobic fermentation pathways. This energy dividend will prove crucial for supporting the complex cellular processes required by larger, more sophisticated organisms. Ozone formation in the upper atmosphere provides Earth with its first effective shield against harmful ultraviolet radiation. As oxygen molecules rise into the stratosphere, solar UV breaks them apart and allows the fragments to recombine into 3-atom ozone molecules. This ozone layer absorbs the most damaging wavelengths of ultraviolet light, creating a protective umbrella that will eventually allow life to colonize the planet's surface without facing immediate radiation damage. The development of this natural sunscreen represents one of life's first successful attempts at planetary-scale environmental engineering. Red bed formations begin appearing in the geological record as iron-rich soils become exposed to atmospheric oxygen for the first time. These distinctive rust-colored sedimentary layers mark the transition from a reducing to an oxidizing surface environment, their red iron oxide minerals serving as geological markers of the oxygenation event. The appearance of red beds in continental sediments provides evidence that oxygen concentrations have reached levels sufficient to penetrate inland environments and alter terrestrial weathering processes. Only took cyanobacteria roughly 1 billion years to poison most of their planetary neighbors through their photosynthetic activities. Only required the patient accumulation of oxygen molecules, 1 photon at a time, to transform Earth's atmosphere from a reducing mixture dominated by methane and hydrogen to an oxidizing environment where complex chemistry becomes possible. Only needed the inadvertent genocide of anaerobic ecosystems to clear ecological space for the aerobic organisms that would eventually inherit the oxygenated world. The evolutionary arms race between oxygen producers and oxygen consumers begins driving biochemical innovations at unprecedented rates. Organisms develop increasingly sophisticated antioxidant systems to cope with oxygen toxicity, including enzymes like superoxide dismutase that neutralize reactive oxygen species before they can cause cellular damage. The competition between photosynthetic oxygen production and biological oxygen consumption creates a dynamic equilibrium that will regulate atmospheric composition for billions of years to come. Seasonal patterns emerge as cyanobacterial productivity responds to changes in solar radiation, temperature, and nutrient availability. The banded iron formations preserve evidence of these ancient rhythms, their alternating layers reflecting periods of high and low photosynthetic activity that may correspond to early seasonal cycles. This biological control over geological processes represents one of the first examples of what modern scientists call the Gaia hypothesis. The idea that life actively regulates planetary environments to maintain conditions suitable for continued biological activity. The mystery of delayed oxygenation haunts researchers studying this crucial transition in Earth's history. Cyanobacteria appear in the fossil record several 100 million years before significant oxygen accumulation begins, suggesting that some mechanism initially prevented atmospheric oxygenation despite active photosynthetic production. Whether this delay resulted from enhanced oxygen consumption by reduced minerals, biological feedback mechanisms, or atmospheric chemistry that we don't fully understand remains one of the major puzzles in the history of Earth's atmosphere. Perhaps the most profound irony of the great oxygenation event lies in its ultimate consequences. The same process that caused Earth's first major mass extinction also created the chemical conditions necessary for complex life to evolve. Without oxygen, the sophisticated cellular machinery that powers animals, plants, and fungi could never have developed. The atmospheric poison produced by ancient cyanobacteria became the essential ingredient for consciousness, intelligence, and the eventual emergence of organisms capable of understanding their own evolutionary history. The eukaryotic revolution begins not with conquest, but with an act of extraordinary cellular hospitality that transforms the fundamental architecture of life on Earth. Somewhere in the ancient oceans, a large prokaryotic cell engulfs a smaller bacterium in what appears to be a routine predatory encounter. But instead of digesting its prey, the host cell allows the captive to survive and reproduce within its cytoplasm. This unlikely partnership between formerly independent organisms creates the first eukaryotic cell, a living system whose complexity exceeds the sum of its constituent parts by orders of magnitude. The captured bacterium gradually transforms into a mitochondrion, surrendering its independence in exchange for a protected environment and steady supply of nutrients. Over countless generations, the endosymbiont transfers most of its genes to the host cell's nucleus, retaining only the essential genetic information needed for its specialized role as the cell's powerhouse. This genetic downsizing represents one of evolution's most dramatic examples of use it or lose it selection, where unnecessary complexity gets systematically eliminated until only the essential functions remain. DNA organization undergoes a revolutionary transformation as genetic material becomes enclosed within a membrane-bound nucleus for the first time in evolutionary history. This nuclear envelope provides protection for the cell's genetic information while creating new opportunities for sophisticated gene regulation through compartmentalized biochemistry. The separation between DNA storage in the nucleus and protein synthesis in the cytoplasm introduces quality control mechanisms that allow much larger genomes to be maintained with acceptable error rates. Linear chromosomes replace the circular DNA molecules that have served prokaryotes successfully for billions of years. These new chromosomal structures require specialized molecular machinery to replicate their ends accurately, leading to the evolution of telomerases and other complex enzymatic systems. The benefits of linear organization become apparent as eukaryotic genomes expand to accommodate thousands of genes encoding proteins with increasingly specialized functions. Introns appear within eukaryotic genes, creating a puzzle that molecular biologists are still working to understand. These non-coding sequences interrupt protein coding regions and must be precisely removed during RNA processing before functional proteins can be synthesized. The splicing machinery required for intron removal rivals the ribosome in complexity. Yet the evolutionary advantage of interrupting genes with non-coding sequences remains mysterious. Perhaps introns facilitate genetic recombination, allow alternative splicing to create protein variants, or simply represent molecular fossils of ancient evolutionary processes. The endomembrane system develops as eukaryotic cells create increasingly sophisticated internal architectures. The endoplasmic reticulum provides specialized environments for protein synthesis and modification, while the Golgi apparatus functions as a molecular post office that sorts and ships cellular products to their proper destinations. These membrane-bound compartments allow different biochemical processes to proceed simultaneously within the same cell without interfering with each other, essentially creating cellular factories with specialized production lines. Sex evolves as eukaryotes discover the genetic benefits of combining DNA from two different individuals rather than simply copying existing genomes during reproduction. Meiosis. The complex cellular process that generates genetically unique gametes requires sophisticated molecular machinery to ensure that each offspring receives exactly one copy of every chromosome. The evolution of sexual reproduction represents one of biology's most significant innovations, creating the genetic diversity that will fuel all subsequent evolutionary advances. The cytoskeleton emerges as eukaryotic cells develop internal scaffolding systems that can actively reshape cellular architecture in response to environmental conditions. Microtubules, composed of tubulin proteins, create rigid supports that maintain cell shape while serving as highways for intracellular transport. Actin filaments provide contractile elements that enable cellular movement and shape changes. This internal framework allows eukaryotic cells to achieve sizes and shapes impossible for prokaryotes while maintaining structural integrity. Phagocytosis develops as some eukaryotes learn to engulf particles and other cells by surrounding them with extensions of their cell membrane. This feeding strategy opens entirely new ecological niches, allowing single-celled predators to consume prey much larger than the dissolved nutrients that sustain prokaryotic metabolism. The ability to internalize other organisms also creates opportunities for additional endosymbiotic events that will lead to even greater cellular complexity. The capture of cyanobacteria by early eukaryotes creates the first photosynthetic eukaryotic cells through a second major endosymbiotic event. These internalized photosynthetic partners gradually evolve into chloroplasts, retaining their own circular DNA and ribosomes while becoming completely dependent on their host cells for survival. The evolution of photosynthetic eukaryotes sets the stage for the evolution of plants and the eventual colonization of terrestrial environments. Only took evolution several 100 million years to discover that cooperation between different organisms could generate capabilities impossible for either partner alone. Only required countless failed attempts at cellular cannibalism before stumbling upon partnerships that proved beneficial to both participants. Only needed the gradual transfer of genes and functions between host and symbiont to create integrated systems that function as unified wholes rather than collections of independent parts. The cellular complexity that emerges during the eukaryotic revolution creates new possibilities for multicellular organization. Individual eukaryotic cells possess the internal machinery needed to specialize for different functions within larger organisms, setting the stage for the evolution of tissues, organs, and eventually complex multicellular bodies. The transition from prokaryotic simplicity to eukaryotic complexity represents one of the most significant leaps in the history of life, comparable to the original emergence of life itself. Gene regulation becomes increasingly sophisticated as eukaryotic cells develop elaborate mechanisms for controlling when and where different genes get expressed. Transcription factors, enhancer sequences, and chromatin modifications create regulatory networks that can coordinate the activities of thousands of genes in response to environmental signals or developmental programs. This regulatory complexity will prove essential for the multicellular organisms that follow, allowing single genomes to specify hundreds of different cell types. The mystery of eukaryotic origins continues to puzzle evolutionary biologists despite decades of research into the molecular details of these ancient cellular mergers. The identity of the host cell that first engulfed the proto-mitochondrion remains uncertain, with different studies suggesting relationships to various groups of archaea or bacteria. The timing of different endosymbiotic events and the sequence in which various eukaryotic features evolved remain active areas of research, with new discoveries regularly revising our understanding of these pivotal events. Perhaps the most profound consequence of the eukaryotic revolution lies not in any specific cellular innovation, but in the demonstration that evolution can create entirely new levels of biological organization through cooperation rather than competition alone. The success of endosymbiosis shows that formerly independent entities can merge to create integrated systems whose capabilities transcend those of their constituent parts. This principle of emergence through cooperation will appear repeatedly throughout subsequent evolutionary history, from multicellular organisms to ecological communities, to human societies that depend on countless forms of biological and technological symbiosis. The iron content in eukaryotic cells increases dramatically compared to their prokaryotic ancestors, reflecting the oxygen-rich environment that makes complex cellular machinery possible. Iron-sulfur clusters become essential components of numerous enzymes involved in energy metabolism, while iron-containing cytochromes facilitate electron transport in both mitochondria and chloroplasts. The chemistry of cellular complexity requires not just sophisticated molecular machinery, but also access to the diverse elemental resources that an oxygenated world provides. Sex and death arrive on Earth as paired innovations that will forever change the trajectory of biological evolution, their simultaneous emergence representing one of evolution's most counterintuitive strategies for long-term survival. The eukaryotic cells that have mastered the art of internal complexity now face a fundamental choice: continue the immortal but inflexible lifestyle of their prokaryotic ancestors, or embrace the creative destruction that comes with sexual reproduction and programmed cellular aging. The decision to abandon individual immortality in favor of genetic mixing proves to be one of the most successful gambles in the history of life. Sexual reproduction begins as simple genetic exchange between single-celled organisms, a primitive version of the bacterial conjugation that allows rapid spread of beneficial mutations through prokaryotic populations. But eukaryotic sex evolves into something far more elaborate and costly, requiring complex cellular machinery to reduce chromosome numbers during meiosis and then restore them during fertilization. The process demands enormous energy investment and creates numerous opportunities for fatal errors. Yet organisms that master sexual reproduction quickly outcompete their asexual relatives in environments where genetic diversity provides survival advantages. Meiosis emerges as one of the most sophisticated cellular processes ever evolved, requiring precise coordination between dozens of different protein complexes to ensure accurate chromosome segregation. Homologous chromosomes must find each other within the crowded nuclear environment, align precisely along their entire length, and exchange genetic material through crossing over before separating cleanly into different daughter cells. A single error in this process can result in offspring with too many or too few chromosomes, usually leading to developmental failure or sterility. The recombination machinery that facilitates genetic crossing over during meiosis creates novel gene combinations that would never arise through mutation alone. Beneficial mutations that occur on the same chromosome can be separated and recombined with other favorable variants, while harmful mutations can be isolated and eliminated more efficiently than in asexual populations. This genetic shuffling accelerates adaptation to changing environments while reducing the accumulation of deleterious mutations that plague asexual lineages over evolutionary time. Gamete formation represents evolution's solution to the mathematical problem of maintaining stable chromosome numbers across generations. If organisms simply combined their complete genomes during reproduction, chromosome numbers would double with each generation until cellular machinery could no longer cope with the genetic excess. Meiosis solves this problem by creating specialized reproductive cells with exactly half the normal chromosome complement, ensuring that fertilization restores the species-specific chromosome number rather than increasing it indefinitely. The evolution of distinct sexes emerges as some organisms develop specialized reproductive roles that optimize either gamete production or gamete delivery. Females invest heavily in producing large, nutrient-rich eggs that can support early embryonic development, while males produce numerous small, mobile sperm that can compete for access to limited egg resources. This sexual dimorphism creates different selective pressures on males and females, driving the evolution of elaborate courtship behaviors and secondary sexual characteristics that will reach spectacular extremes in later evolutionary periods. Programmed cell death evolves alongside sexual reproduction as multicellular organisms learn to eliminate aging or damaged cells before they can threaten the health of the entire organism. Apoptosis. The controlled cellular suicide process requires sophisticated molecular machinery to dismantle cellular components in an orderly fashion while preventing the release of toxic contents that could damage neighboring cells. This seemingly wasteful process proves essential for maintaining tissue health and preventing the uncontrolled cellular proliferation that characterizes cancer. The paradox of aging emerges as organisms that possess the molecular machinery for cellular immortality nevertheless evolve mechanisms that limit their own lifespan. Telomeres. The protective DNA sequences at chromosome ends progressively shorten with each cell division until they trigger senescence pathways that halt further cellular reproduction. This cellular aging clock appears to result from evolutionary trade-offs between cancer prevention and tissue maintenance, with natural selection favoring mechanisms that prevent runaway cellular growth even at the cost of eventual organismal death. Only took evolution several hundred million years to discover that immortal individuals make mortal species. Only required the counterintuitive leap from preserving existing genetic combinations to constantly creating new ones through sexual recombination. Only needed the recognition that individual death becomes advantageous when it accelerates genetic innovation at the population level, allowing species to adapt more rapidly to changing environmental conditions than immortal but genetically static lineages. The cost-benefit analysis of sexual reproduction continues to puzzle evolutionary biologists studying modern organisms. Sexual reproduction reduces an individual's genetic contribution to future generations by 50% compared to asexual reproduction, while requiring substantial energy investments in mate finding, courtship, and gamete production. Despite these costs, sexual reproduction dominates among complex eukaryotes, suggesting that the benefits of genetic recombination outweigh the disadvantages in most ecological contexts. Senescence patterns vary dramatically among different organism groups, with some species aging rapidly after a single reproductive event, while others maintain reproductive capacity for centuries. The molecular mechanisms underlying these differences involve complex interactions between cellular repair systems, metabolic rates, and environmental stresses that determine how quickly aging damage accumulates relative to repair capabilities. Understanding these patterns provides insights into the fundamental trade-offs that shape life history evolution. The Red Queen hypothesis emerges from observations that sexual reproduction appears most advantageous in environments where organisms face rapidly evolving threats such as parasites and pathogens. In these dynamic ecological contexts, the genetic diversity generated by sexual reproduction allows host populations to stay ahead of parasites that continuously evolve new ways to exploit them. Asexual organisms, lacking this genetic flexibility, often succumb to specialized parasites that can rapidly spread through genetically uniform populations. Chromosomal sex determination systems evolve as some organisms develop genetic mechanisms that ensure balanced production of males and females. The evolution of sex chromosomes requires complex genetic rearrangements that prevent recombination between male-determining and female-determining regions, leading to the accumulation of genetic differences that eventually create distinct X and Y chromosomes. These sex determination systems become increasingly elaborate as organisms develop mechanisms to compensate for gene dosage differences between males and females. The molecular machinery of sexual reproduction reveals stunning biochemical complexity beneath the apparent simplicity of genetic exchange. Proteins involved in chromosome pairing, recombination, and segregation show remarkable conservation across diverse eukaryotic lineages, suggesting that the basic mechanisms of meiosis evolved early and proved so successful that subsequent evolution refined rather than replaced them. The universality of sexual reproduction among complex organisms testifies to its fundamental importance for long-term evolutionary success. The mystery of sex ratios continues to intrigue researchers studying the evolutionary forces that determine the proportion of males and females in different populations. Fisher's principle suggests that equal sex ratios should evolve because rare sexes enjoy reproductive advantages that restore balance. Yet many organisms deviate from equality in ways that reflect local ecological conditions or genetic constraints. Environmental sex determination, where temperature or other factors influence offspring sex ratios, adds additional complexity to our understanding of how reproductive strategies evolve. Perhaps the most profound philosophical implication of sexual reproduction lies in its demonstration that individual sacrifice can serve collective benefit in evolutionary contexts. The willingness of organisms to halve their genetic contribution to future generations in order to participate in sexual reproduction shows that natural selection can favor behaviors that appear individually costly but prove beneficial at higher levels of biological organization. This principle of multi-level selection will become increasingly important as evolution proceeds toward greater complexity and cooperation among living systems. The first multicellular experiments unfold in ancient seas as single-celled organisms begin testing the revolutionary concept that cooperation might prove more advantageous than competition. Simple colonial arrangements emerge where identical cells remain loosely associated after division, sharing resources and facing environmental challenges as unified groups rather than isolated individuals. These early cellular federations represent tentative steps toward true multicellularity, exploratory ventures that will eventually transform Earth's biosphere from a collection of microscopic individuals into complex ecosystems dominated by large, integrated organisms. Volvox colonies demonstrate one of evolution's most elegant solutions to the transition from unicellular to multicellular life. These spherical aggregations contain thousands of flagellated cells arranged in a hollow ball, their coordinated beating creating currents that propel the entire colony through the water like a living green marble. Individual cells within the colony begin showing the first signs of functional specialization, with most cells devoted to swimming and feeding, while a small subset develops into reproductive cells capable of producing new colonies. This division of labor represents a crucial conceptual leap from cellular equality to cellular hierarchy. Cell adhesion molecules evolve as multicellular organisms develop molecular mechanisms for maintaining physical connections between individual cells. These protein complexes create specific binding sites that allow cells of the same type to recognize and attach to each other while rejecting foreign cells that might disrupt tissue integrity. The evolution of selective cellular adhesion provides the foundation for tissue formation, allowing groups of specialized cells to maintain their organization despite the constant physical stresses of growth, movement, and environmental turbulence. Extracellular matrices begin appearing as multicellular organisms secrete structural proteins and polysaccharides that create supportive frameworks around and between their constituent cells. Collagen, elastin, and other fibrous proteins form networks that provide mechanical strength while maintaining flexibility, essentially creating biological scaffolding that can support increasingly large and complex cellular arrangements. These extracellular structures also serve as highways for cellular communication, carrying chemical signals between distant parts of developing organisms. Gap junctions evolve as adjacent cells develop direct cytoplasmic connections that allow rapid exchange of small molecules and ions. These intercellular channels create electrical and chemical continuity between cells that would otherwise remain isolated by their plasma membranes, enabling coordinated responses to environmental stimuli across entire tissues. The ability to share metabolites, signaling molecules, and electrical currents through gap junctions represents one of the first steps toward the integrated physiological systems that characterize complex multicellular organisms. Cell differentiation emerges as identical cells within multicellular organisms begin expressing different subsets of their shared genome to create functionally distinct cell types. This process requires sophisticated gene regulatory networks that can maintain stable patterns of gene expression through multiple cell divisions while responding appropriately to developmental signals. The evolution of cellular differentiation allows single organisms to contain multiple specialized cell types, each optimized for specific functions within the larger biological community. Apoptosis, the programmed cell death mechanism that evolved alongside sexual reproduction, finds new applications in multicellular development as organisms learn to use controlled cell death to sculpt complex three-dimensional structures. The elimination of cells in specific patterns creates spaces between developing structures, removes obsolete embryonic tissues, and eliminates cells that fail to integrate properly into developing organ systems. This creative use of destruction proves essential for generating the complex anatomical arrangements that characterize advanced multicellular organisms. Size advantages begin manifesting as multicellular organisms explore ecological niches unavailable to single-celled competitors. Larger organisms can maintain more stable internal environments, store greater energy reserves, and access food sources too large for unicellular predators. The ability to grow to substantial sizes while maintaining cellular organization creates opportunities for internal specialization that would be impossible within the confines of single-cell architecture. Only took evolution several 100 million years to discover that cellular committees could outperform individual cellular entrepreneurs. Only required countless experiments with colonial living before stumbling upon the organizational principles that make true multicellularity possible. Only needed the gradual evolution of molecular mechanisms that allow thousands or millions of cells to function as unified wholes rather than collections of competing individuals. Communication networks develop as multicellular organisms create increasingly sophisticated systems for coordinating activities across their cellular populations. Chemical signaling pathways allow cells to exchange information about local conditions, developmental status, and resource availability, while electrical signals provide rapid communication channels for time-sensitive responses. These communication systems represent the earliest versions of the nervous and endocrine systems that will eventually coordinate the activities of complex animals. Developmental programs emerge as multicellular organisms evolve genetic mechanisms that can reliably produce complex three-dimensional structures from simple cellular starting points. Master control genes begin appearing that can switch entire developmental pathways on or off, creating cascading regulatory networks that transform single fertilized eggs into organisms containing dozens of different cell types arranged in precise anatomical patterns. The evolution of robust developmental programs allows multicellular organisms to maintain their complex organization across generations despite the inherent randomness of cellular processes. Tissue boundaries form as different cell types organize themselves into distinct layers and compartments within developing organisms. The creation of these biological borders requires sophisticated molecular machinery that can maintain separation between different tissue types while allowing controlled exchange of materials and information across boundaries. Basement membranes, composed of specialized extracellular matrix proteins, provide structural barriers that help organize tissues while serving as platforms for cellular migration and signaling. The emergence of polarity represents another crucial innovation in multicellular organization. As organisms develop mechanisms for creating and maintaining directional differences in their body plans, head-tail gradients, dorsal-ventral distinctions, and inside-outside orientations require complex molecular machinery that can establish and maintain spatial information across large cellular populations. These polarity systems provide the organizational framework necessary for creating bodies with distinct front and back ends, top and bottom surfaces, and internal and external environments. Stem cell populations evolve as multicellular organisms develop renewable sources of undifferentiated cells that can replace damaged or aging tissues throughout the organism's lifetime. These cellular reservoirs maintain their capacity for division and differentiation while producing specialized descendant cells that integrate into existing tissues. The evolution of stem cell systems allows complex multicellular organisms to maintain and repair their sophisticated cellular architectures despite the inevitable wear and tear of biological existence. Growth regulation becomes increasingly important as multicellular organisms develop mechanisms for controlling their overall size and proportions. Tumor suppressor genes and growth factor signaling pathways evolve to prevent uncontrolled cellular proliferation while ensuring that different body parts grow at appropriate rates relative to each other. The balance between cellular proliferation and growth control determines whether multicellular organisms develop into well-proportioned, functional entities or collapse into disorganized masses of competing cellular lineages. The mystery of multicellular origins continues to challenge researchers studying the transition from unicellular to multicellular life. While colonial organisms like Volvox provide insights into possible intermediate stages, the fossil record preserves few direct traces of the earliest multicellular experiments. The molecular mechanisms that enable multicellular organization appear so complex that their evolution seems to require numerous coordinated innovations. Yet, multicellularity has evolved independently multiple times in different lineages, suggesting that the transition may be more accessible than its complexity suggests. Perhaps the most remarkable aspect of early multicellular evolution lies in its demonstration that individual cells can surrender their autonomy to become components of larger, more complex systems. This transition requires fundamental changes in cellular behavior, from competing with neighbors for resources to cooperating in ways that benefit the larger organism even when they disadvantage individual cells. The success of multicellular organisms shows that evolution can create levels of organization that transcend their constituent parts, establishing principles of biological cooperation that will eventually enable the evolution of societies, ecosystems, and technological civilizations. The Ediacaran gardens spread across the ancient seafloor like living quilts, their strange geometries defying every category that paleontologists will later attempt to impose upon them. These enigmatic organisms emerged during Earth's first experiment with large, complex multicellular life, their soft bodies reaching sizes that dwarf anything the microbial world has previously produced. Dickinsonia grows to dinnerplate dimensions while maintaining the simple architecture of a segmented mat. Its body plan so unlike any modern organism that scientists debate whether it represents an animal, plant, fungus, or something else entirely that no longer exists on Earth. The Avalon assemblage preserves the earliest glimpses of these peculiar communities in rocks that formed on deep ocean floors where sunlight never penetrated. Charnia and its relatives anchor themselves to the seafloor with hold-fast structures, their frond-like bodies extending into the water column to filter nutrients from the surrounding medium. These rangeomorphs display fractal growth patterns where the same branching motifs repeat at multiple scales, creating organisms that resemble mathematical equations more than recognizable life forms. Their feeding strategies remain mysterious, as they lack obvious mouths, digestive systems, or any of the anatomical features associated with conventional animal nutrition. Cambrian represents one of the few Ediacaran organisms that shows clear evidence of directed movement across the sea floor. Fossil trails, preserved alongside its body impressions, suggest a mobile lifestyle involving systematic exploitation of microbial mats that covered the ocean bottom like vast bacterial carpets. The creature's asymmetrical body plan and associated trace fossils hint at the presence of a muscular foot similar to those found in modern mollusks, providing tantalizing evidence for the early evolution of animal-like locomotion and feeding behaviors. The preservation of these soft-bodied organisms requires extraordinary circumstances that rarely occur in the geological record. Most Ediacaran fossils form through a process called death mask preservation, where organisms become buried in fine sediments that harden around their bodies before decay can eliminate all traces of their existence. The resulting impressions capture anatomical details with remarkable fidelity, preserving the quilted textures, segmented patterns, and branching structures that characterize these ancient life forms. Tribrachidium displays threefold radial symmetry unlike any modern animal group. Its disc-shaped body adorned with curved ridges that spiral outward from a central hub. This unusual geometry suggests feeding or reproductive strategies that have no modern analogues, representing experiments in body plan organization that evolution later abandoned in favor of the bilateral symmetry that dominates complex animal life. The threefold symmetry appears in several different Ediacaran genera, suggesting that this organizational pattern proved temporarily successful before disappearing from the fossil record. The ecological relationships within Ediacaran communities remain largely mysterious, as these organisms show little evidence for the predator-prey interactions that structure modern ecosystems. Most Ediacaran organisms appear to have been filter feeders, absorbing dissolved organic matter or capturing microscopic particles from the water column rather than consuming other large organisms. This peaceful coexistence may reflect the absence of effective predatory strategies or defensive mechanisms, creating ecosystems based on resource partitioning rather than direct competition. Microbial mats continue playing crucial roles in Ediacaran ecosystems, providing both habitat structure and nutrient sources for the larger organisms that live above them. These bacterial and algal communities create complex three-dimensional landscapes on the sea floor, their photosynthetic and chemosynthetic activities supporting food webs that extend upward into the water column. The interaction between Ediacaran organisms and their microbial substrates represents one of the earliest examples of large organisms depending on microbial communities for survival. The Ediacaran biota occupies a unique position in evolutionary history, representing Earth's first sustained experiment with large, complex multicellular life while remaining completely distinct from all subsequent animal communities. These organisms achieve remarkable size and morphological complexity without developing the hard shells, sophisticated sensory systems, or active predatory behaviors that will characterize later animal radiations. Their success demonstrates that alternative pathways to biological complexity exist beyond those that ultimately led to modern animal designs. Only took evolution several 100 million years to move beyond microscopic cellular arrangements and begin exploring the possibilities inherent in meter-scale multicellular architectures. Only required the development of mechanisms for maintaining cellular coordination across unprecedented size ranges while experimenting with body plans that would never again appear in the fossil record. only needed the temporary absence of effective predators to allow these peaceful giants to flourish in ecosystems unlike any that would follow. Body plan innovations during the Ediacaran period include the first experiments with bilateral symmetry, radial organization, and complex branching patterns that foreshadow later evolutionary developments. Some organisms display clear head-tail differentiation and left-right symmetry that anticipate the bilateral body plans of most modern animals, while others explore radial symmetries that will later prove successful in cnidarians and echinoderms. The diversity of organizational experiments suggests that early multicellular life tested numerous architectural solutions before settling on the designs that dominate modern ecosystems. The absence of mineralized skeletons among Ediacaran organisms creates preservation challenges that limit our understanding of their biology and ecology. Unlike later periods when shells, bones, and other hard parts create abundant fossil records, Ediacaran life forms depend entirely on soft tissue preservation that occurs only under exceptional circumstances. This preservational bias means that many Ediacaran organisms may remain unknown, while others are represented by incomplete specimens that reveal only partial information about their anatomy and lifestyle. Chemical fossil evidence suggests that some Ediacaran organisms may have possessed biochemical pathways unlike those found in any modern life forms. Styrene molecules preserved in rocks from this period include compounds that modern organisms cannot synthesize, suggesting the presence of metabolic capabilities that disappeared during later evolutionary transitions. These molecular fossils provide tantalizing hints about the physiological diversity of early complex life, while raising questions about why certain biochemical innovations failed to persist into modern ecosystems. Environmental conditions during the Ediacaran period favor the preservation and success of soft-bodied organisms in ways that later environmental changes will not support. Ocean chemistry remains relatively stable, with lower oxygen concentrations that may have inhibited the evolution of active predators while providing sufficient oxygen to support large, complex organisms. The absence of bioturbation by burrowing animals allows delicate seafloor communities to persist without disruption, creating ecological conditions that will never again occur in Earth's history. The mystery of Ediacaran extinction remains one of paleontology's most intriguing puzzles, as these successful and diverse organisms largely disappear from the fossil record just before the Cambrian Explosion brings familiar animal groups into prominence. Whether the Ediacaran biota succumb to environmental changes, competition from new animal groups, or predation pressure from emerging carnivorous lifestyles continues to generate scientific debate. The transition from Ediacaran to Cambrian faunas represents one of the most dramatic turnovers in the history of life, marking the end of Earth's first experiment with large-scale multicellular complexity. Perhaps the most profound lesson of the Ediacaran period lies in its demonstration that successful biological complexity can take forms completely different from those familiar in modern ecosystems. These organisms achieved sizes and organizational sophistication comparable to many modern animals, while following developmental and ecological strategies that have no contemporary parallels. Their existence expands our understanding of life's possibilities while reminding us that evolution's creativity extends far beyond the solutions that ultimately proved most persistent across geological time. The Cambrian Explosion begins not with violence, but with the patient construction of Earth's first hard parts, as organisms discover the transformative power of controlled mineral precipitation within their own tissues. In shallow seas warmed by an increasingly powerful sun, creatures that have spent eons as soft-bodied wanderers across seafloor meadows begin experimenting with calcium carbonate, silica, and phosphate compounds that can be secreted and shaped into protective shells, cutting tools, and structural supports. The innovation of biomineralization unleashes an arms race that will forever change the rules of biological interaction, transforming peaceful Ediacaran communities into battlegrounds where predation and defense drive evolutionary innovation at unprecedented rates. Claudina represents one of the earliest attempts at skeletal construction. Its simple tube-shaped shell providing protection for a soft-bodied inhabitant whose identity remains mysterious. These primitive shells stack like geological cylinders, their walls perforated by holes that may have allowed feeding tentacles to extend into the surrounding water. The appearance of repair scars on some Claudina shells provides the first direct evidence of predation attempts in the fossil record, marking the moment when Earth's organisms began actively trying to consume each other rather than simply competing for dissolved nutrients. Small shelly fossils proliferate throughout the early Cambrian as numerous organism groups independently discover the advantages of hard protective coverings. Archaeocyaths construct reef-like structures from calcium carbonate, their porous walls creating three-dimensional habitats that shelter smaller organisms while filtering nutrients from seawater. Hyoliths develop cone-shaped shells with hinged opercula that can seal their openings like biological bank vaults, while tomatids experiment with scale-like armor plates that anticipate the more sophisticated defensive systems of later animals. Trilobites emerge as the Cambrian's most successful arthropods, their segmented bodies protected by flexible exoskeletons that can be molted to accommodate growth while maintaining protective integrity. These marine arthropods develop compound eyes containing thousands of individual lenses, creating visual systems capable of detecting movement, recognizing shapes, and navigating complex three-dimensional environments. The evolution of sophisticated vision transforms ecological interactions by allowing predators to hunt more effectively while enabling prey species to detect and escape approaching threats. Anomalocaridids grow to unprecedented sizes as Cambrian seas produce the first large predators capable of actively hunting other macroscopic organisms. Anomalocaris reaches lengths exceeding 2 meters. Its grasping arms equipped with spines that can capture and manipulate prey, while its circular mouth contains rings of teeth adapted for processing hard-shelled victims. These apex predators patrol Cambrian reefs like prehistoric sharks, their presence creating intense selective pressure for improved defensive strategies among potential prey species. Brachiopods establish themselves as dominant filter feeders, their hinged shells protecting soft tissues while allowing controlled water flow for feeding and respiration. These organisms anchor themselves to hard substrates using flexible stalks called pedicles, creating the first sessile communities that transform seafloor rocks into complex three-dimensional habitats. The success of brachiopods demonstrates that effective defense can be as evolutionarily advantageous as aggressive predation, allowing organisms to thrive by perfecting protective strategies rather than pursuing carnivorous lifestyles. The Burgess Shale preserves extraordinary detail of Cambrian soft tissue anatomy, revealing the full complexity of organisms whose hard parts alone would suggest much simpler biology. Weywaxia displays a coat of spines and scales that creates a defensive barrier around its soft body, while Opabinia extends a bizarre proboscis tipped with a grasping claw that seems designed for extracting small prey from crevices. These exceptional fossils demonstrate that Cambrian innovation extended far beyond shell construction to include sophisticated soft tissue specializations. Only took evolution a few tens of millions of years to transform Earth's biosphere from communities of peaceful filter feeders into ecosystems dominated by predator-prey dynamics. Only required the discovery of biomineralization to trigger an arms race that would drive animal diversification for the next 500 million years. Only needed the invention of eyes, teeth, claws, and armor to create the first recognizably modern ecological interactions based on hunting, hiding, and competitive exclusion. Trace fossils multiply dramatically during the Cambrian as organisms develop new capabilities for movement and substrate manipulation. Burrow systems become increasingly complex as animals learn to excavate three-dimensional networks within seafloor sediments, creating protected microenvironments while accessing food resources buried below the surface. The evolution of effective burrowing strategies allows organisms to exploit entirely new ecological niches while creating bioturbation patterns that fundamentally alter seafloor chemistry and structure. Biomineralization pathways evolve independently in multiple organism groups, suggesting that the chemical prerequisites for hard part construction become widely available during the early Cambrian. The precipitation of calcium carbonate requires precise control over pH, ion concentrations, and organic matrix proteins that guide crystal formation. Yet numerous unrelated lineages master these complex biochemical processes within relatively short geological intervals. This convergent evolution of skeletal construction demonstrates that environmental conditions strongly favor organisms capable of producing hard defensive structures. Reef ecosystems emerge as calcifying organisms begin constructing three-dimensional habitat structures that persist beyond the lifespans of their builders. Archaeocyathene reefs create vertical relief on otherwise flat sea floors, providing attachment sites for sessile organisms while creating current patterns that concentrate nutrients and remove waste products. These early reef communities demonstrate the ecosystem engineering capabilities that will later allow coral reefs to become among Earth's most productive and diverse biological communities. The evolution of jointed appendages allows arthropods to develop sophisticated locomotory capabilities that enable both predatory pursuit and evasive maneuvering. Trilobite legs contain multiple joints that can be coordinated to produce walking, swimming, and digging movements, while specialized appendages evolve for feeding, sensing, and reproductive functions. This appendage diversification creates the morphological foundation for the arthropod success that will make them the most diverse animal group on Earth. Digestive system specialization accompanies the shift toward carnivorous lifestyles as predatory animals develop anatomical structures optimized for capturing, processing, and extracting nutrients from other organisms. Gut regions become differentiated into specialized compartments for mechanical breakdown, chemical digestion, and nutrient absorption, while mouth parts evolve into increasingly sophisticated tools for prey manipulation. These digestive innovations allow predators to extract maximum energy from their prey while minimizing the time and effort required for food processing. Defensive strategies proliferate as prey organisms develop increasingly elaborate mechanisms for avoiding predation. Shell thickening, spine development, and behavioral modifications like burrowing represent alternative solutions to the predation problem, each involving different trade-offs between protection and other biological functions like growth, reproduction, and feeding efficiency. The diversity of defensive innovations demonstrates that multiple evolutionary pathways can lead to successful predator avoidance. The mystery of the Cambrian Explosion's timing continues to puzzle researchers studying the environmental and genetic factors that enabled this remarkable diversification event. Whether the explosion resulted from reaching critical oxygen thresholds, the evolution of key developmental genes, or ecological feedback loops between predators and prey remains actively debated. The rapidity of morphological innovation during this period suggests that fundamental biological constraints were overcome, allowing evolution to explore morphological possibilities that had previously been inaccessible. Perhaps the most significant long-term consequence of the Cambrian explosion lies in its establishment of ecological patterns that continue to structure biological communities today. The predator-prey dynamics that emerged during this period created selective pressures for improved sensory systems, locomotory capabilities, and defensive mechanisms that drive ongoing evolutionary innovation. The arms race between attack and defense that began in Cambrian seas continues to generate biological diversity through mechanisms of escalation and counter-escalation that show no signs of reaching evolutionary equilibrium. Trilobites inherit the ancient seas with the patient confidence of creatures perfectly adapted to their liquid world. Their compound eyes scanning seafloor landscapes that stretch beyond every horizon like vast underwater prairies. For nearly 300 million years, these segmented arthropods will dominate marine ecosystems with a success that makes their eventual extinction seem impossible to contemplate. Their flexible exoskeletons accommodate growth through molting cycles that leave behind countless shed shells, destined to become the most abundant fossils in Paleozoic rocks, testimony to populations that numbered in the billions across every ocean basin. The diversity of trilobite body plans rivals that of any major animal group, with over 20,000 described species exploring every conceivable variation on the arthropod architectural theme. Agnostus develops a pill-bug lifestyle, rolling into a perfect sphere when threatened while maintaining the ability to unfurl and scuttle away when danger passes. Isotelus grows to dinner plate proportions, its broad, flattened form adapted for gliding over soft sediments like a living snowshoe that distributes weight across maximum surface area. Dicronurus sprouts elaborate spines that transform its exoskeleton into a walking cactus, each projection serving as both defensive weapon and stabilizing anchor in strong currents. Compound eyes reach unprecedented sophistication in trilobite visual systems, with some species developing thousands of individual lenses arranged in geometrically perfect arrays that maximize light-gathering efficiency while minimizing optical distortion. Phacops constructs its lenses from pure calcite crystals oriented along precise crystallographic axes that eliminate double images through the mineral's natural optical properties. These biological cameras can detect polarized light, track moving objects, and function effectively in the dim conditions of deep seafloor environments where photosynthesis becomes impossible. Enrollment behavior evolves as trilobites perfect the art of defensive ball formation, their segmented exoskeletons folding along predetermined lines that bring head and tail together in seamless protective spheres. The mechanical precision required for this transformation demands exact coordination between dozens of body segments, each equipped with specialized joints and locking mechanisms that engage automatically when the animal contracts its longitudinal muscles. Enrolled trilobites can survive predator attacks, transport by strong currents, and burial by sediment while maintaining minimal energy expenditure. Filter feeding strategies allow many trilobite species to exploit the vast resources of suspended organic matter that drift through ancient oceans like nutritious snow. Specialized appendages create feeding currents that draw water through elaborate filtering systems constructed from modified gill branches and setae arrays. These biological sieves can extract particles ranging from bacteria to small multicellular organisms, allowing trilobites to harvest productivity from the entire water column rather than depending solely on seafloor resources. Trace fossils preserve evidence of trilobite behavior patterns that reveal complex interactions with their physical and biological environments. Cruziana tracks record the systematic meandering of trilobites across seafloor surfaces as they plowed through sediments in search of buried food resources. These feeding traces follow mathematical patterns that optimize energy expenditure while maximizing the probability of encountering prey, demonstrating that even ancient arthropods possessed sophisticated behavioral algorithms for resource exploitation. Trilobite reproduction involves elaborate mating rituals preserved as fossils, showing multiple individuals arranged in specific geometric patterns that suggest coordinated spawning behaviors. Some species aggregate in vast numbers during reproductive seasons, their molted exoskeletons creating fossil accumulations that record ancient breeding grounds. The development of specialized clasping appendages in male trilobites indicates that copulation involved direct physical contact rather than the broadcast spawning strategies employed by many marine invertebrates. Only took trilobites a few tens of millions of years to explore nearly every possible marine ecological niche, from shallow tidal pools to abyssal depths where sunlight never penetrates. Only required the perfection of arthropod segmentation and jointed appendages to create body plans flexible enough to adapt to environments ranging from tropical reefs to polar seas. Only needed the development of compound vision and sophisticated behaviors to become the first animals capable of complex interactions with three-dimensional marine environments. Developmental plasticity allows trilobite populations to respond rapidly to environmental changes through phenotypic modifications that occur within individual lifespans. Temperature fluctuations influence growth rates and final body sizes, while food availability affects the development of specialized feeding appendages and digestive systems. This developmental flexibility provides trilobite lineages with resilience against short-term environmental perturbations while maintaining genetic capacity for longer-term evolutionary responses. Biomineralization reaches new levels of sophistication as trilobites perfect the art of constructing exoskeletons that balance protection against flexibility, weight, and metabolic cost. Calcium carbonate and calcium phosphate minerals are precisely organized within organic matrices that control crystal orientation, size, and mechanical properties. The resulting composite materials achieve strength-to-weight ratios that exceed many modern engineering materials while maintaining the self-repair capabilities necessary for biological systems. Geographic distribution patterns reveal trilobite dispersal capabilities that span entire ocean basins, with some species maintaining genetic continuity across thousands of kilometers of open water. Larval stages must possess remarkable longevity and navigational abilities to achieve such widespread distributions, suggesting sophisticated adaptations for long-distance oceanic travel. The cosmopolitan distribution of many trilobite genera demonstrates that Paleozoic oceans functioned as highways for biological exchange rather than barriers to dispersal. Predator-prey relationships structure trilobite communities through complex networks of attack and defense strategies that drive ongoing evolutionary innovation. Anomalocaridid predators develop increasingly sophisticated prey-handling capabilities, while trilobites respond with improved enrollment mechanisms, defensive spines, and escape behaviors. These evolutionary arms races create selective pressures that maintain rapid morphological evolution within trilobite lineages throughout their geological history. Ecological specialization reaches remarkable extremes as different trilobite lineages adapt to increasingly narrow environmental niches. Pelagic species develop streamlined body forms and enlarged eyes for life in open water, while benthic specialists perfect burrowing capabilities that allow exploitation of infaunal resources. Some lineages become specialized scavengers that feed exclusively on organic matter settling from the water column, while others develop predatory lifestyles that target specific prey organisms. The mystery of trilobite decline becomes increasingly apparent as Paleozoic ecosystems undergo fundamental changes that gradually erode the ecological foundations of arthropod dominance. The evolution of jawed fish introduces new predatory pressures that trilobite defensive strategies cannot effectively counter, While competition from emerging arthropod groups like early crustaceans challenges trilobite monopolies in traditional ecological niches, climate changes associated with continental drift and ice age cycles create environmental instabilities that exceed trilobite adaptive capabilities. Mass extinction events punctuate trilobite evolutionary history, with episodes of catastrophic diversity loss that eliminate entire families and orders within geologically brief intervals. The Ordovician-Silurian extinction removes many early trilobite lineages, while later extinction events continue the pattern of episodic diversity reduction that eventually drives these once dominant arthropods toward their final disappearance during the Permian extinction. Each survival episode leaves trilobite faunas increasingly impoverished and ecologically marginalized. Fossil preservation quality in trilobite-bearing rocks often exceeds that found in younger deposits, with soft tissue details occasionally preserved alongside typical exoskeleton remains. Pyritization processes can preserve muscle fibers, digestive systems, and even embryonic development stages that provide unprecedented insights into trilobite anatomy and physiology. These exceptional fossils reveal the full complexity of trilobite biology while demonstrating that their apparent simplicity reflects preservational biases rather than actual anatomical limitations. Perhaps the most poignant aspect of trilobite evolutionary history lies in their demonstration that even the most successful biological designs eventually succumb to environmental changes that exceed their adaptive capabilities. These arthropods dominated marine ecosystems for longer than mammals have existed on land, yet their ultimate extinction shows that evolutionary success provides no guarantee against future failure. The trilobite legacy persists in their arthropod relatives, whose continued success demonstrates that fundamental body plans can survive even when particular lineages face extinction, ensuring that biological innovations persist beyond the fate of any individual species. Life ventures onto barren land with the tentative steps of organisms evolved for an entirely aquatic existence, their first terrestrial experiments requiring fundamental reimagining of every biological process that water had previously facilitated. The ancient continents stretch like vast sterile beaches beneath skies that rain down ultraviolet radiation unfiltered by protective ozone layers. Their rocky surfaces scoured by winds that carry only dust and volcanic ash rather than the spores and seeds that will later make terrestrial dispersal possible. In this alien landscape, simple organisms begin the patient work of creating soil, atmosphere, and eventually entire ecosystems from nothing but rock, rain, and the accumulated wisdom of billions of years of aquatic evolution. Cyanobacterial mats pioneer the terrestrial frontier as photosynthetic microbes discover that wet rock surfaces can support metabolism just as effectively as shallow marine environments. These bacterial carpets spread across coastal zones where tidal action provides regular moisture while exposing communities to the novel challenges of atmospheric exposure. The transition from perpetual immersion to cyclic wetting and drying requires sophisticated cellular mechanisms for surviving dehydration, including specialized polymers that protect cellular machinery during dry periods and rapid rehydration systems that restore metabolic activity when water returns. Lichens emerge from the most unlikely partnership in biological history, as fungi and photosynthetic partners forge alliances that neither organism could survive alone in terrestrial environments. The fungal component provides structural support and water retention capabilities while creating chemical environments that dissolve nutrients from bare rock surfaces. The photosynthetic partner, whether cyanobacterium or alga, converts atmospheric carbon dioxide into organic compounds that nourish both members of the alliance. This symbiotic cooperation creates Earth's first successful terrestrial organisms while demonstrating that colonization of novel environments often requires collaboration between unrelated biological systems. Rock weathering accelerates dramatically as biological processes begin supplementing the purely physical and chemical forces that have slowly eroded continental surfaces throughout geological history. Lichen acids dissolve minerals that would otherwise resist breakdown for millions of years, while fungal networks penetrate rock fractures and create expansive pressure systems that fracture even resistant substrates. The organic acids produced by these pioneer organisms alter soil chemistry in ways that make mineral nutrients available to subsequent colonists while creating buffering systems that moderate pH fluctuations. Simple plants make their terrestrial debut as aquatic algae, develop increasingly sophisticated mechanisms for surviving exposure to air, drought, and intense solar radiation. Charophytes, the green algae most closely related to land plants, already possess many of the cellular features necessary for terrestrial survival, including cellulose cell walls that provide structural support and specialized reproductive structures that protect developing gametes from environmental stresses. The transition from water to land requires relatively few additional innovations beyond those already present in advanced aquatic plant lineages. Bryophytes represent the first true land plants, their simple architecture revealing both the possibilities and limitations of early terrestrial plant design. Mosses, liverworts, and hornworts lack the sophisticated plumbing systems that will later allow plants to grow to massive sizes, instead depending on direct absorption of water through their surfaces and simple diffusion to transport nutrients throughout their small bodies. This architectural simplicity restricts bryophytes to moist environments where water remains readily available, but also allows them to colonize habitats where more complex plants cannot establish themselves. Spore dispersal mechanisms evolve as terrestrial plants develop strategies for spreading their offspring across landscape scales without depending on water currents that facilitate reproduction in aquatic environments. Wind dispersal requires specialized spore wall chemistry that protects genetic material during extended atmospheric transport while maintaining viability for germination when suitable habitats are encountered. The evolution of resistant spore coats represents one of the crucial innovations that allows terrestrial reproduction to succeed despite the harsh conditions of aerial environments. Soil formation begins accelerating as plant and fungal activities create the first stable terrestrial substrates capable of supporting complex biological communities. Root systems, even in simple early plants, create networks of channels that allow water infiltration and gas exchange while physically binding mineral particles into coherent soil structures. Organic matter from dead plant tissues provides the chemical foundation for soil ecosystems, creating nutrient cycles that will eventually support vast terrestrial forests and grasslands. Only took life several 100 million years to recognize that land surfaces offered vast unexploited resources for organisms capable of surviving atmospheric exposure. Only required the patient evolution of cellular mechanisms for surviving dehydration, obtaining nutrients from mineral substrates, and reproducing without aquatic media. Only needed the development of symbiotic partnerships that could combine the capabilities of different organisms into integrated systems capable of terrestrial success. Vascular tissue evolution represents a breakthrough that will eventually allow plants to achieve sizes that dwarf their aquatic ancestors. The development of specialized cells for water and nutrient transport creates internal plumbing systems that can move materials across distances impossible for simple diffusion-based transport. Xylem tissue provides both mechanical support and water transport capabilities, while phloem creates distribution networks for the organic compounds produced by photosynthesis. These innovations lay the groundwork for the complex plant architectures that will eventually dominate terrestrial landscapes. Stomatal regulation emerges as land plants develop sophisticated mechanisms for controlling gas exchange between their internal tissues and the atmosphere. These microscopic pores allow carbon dioxide uptake for photosynthesis while minimizing water loss through transpiration, creating a delicate balance between metabolic needs and conservation requirements. The evolution of stomatal control represents one of the first examples of sophisticated environmental sensing and response systems in terrestrial organisms. Protective waxy coatings develop as land plants perfect the chemistry of water retention through specialized lipid layers that reduce evaporation from exposed surfaces. Cuticles composed of cutin and other waterproof compounds create barriers that allow plants to maintain internal water content even in dry atmospheric conditions. The thickness and composition of these protective layers can be adjusted in response to environmental conditions, providing flexible defense against desiccation stress. Mycorrhizal associations evolve as plant roots form increasingly intimate partnerships with soil fungi that expand their effective surface area for nutrient absorption. These symbiotic relationships allow plants to access phosphorus, nitrogen, and other essential elements from soil volumes, far exceeding those reachable by root systems alone. In exchange, plants provide fungal partners with carbohydrates produced through photosynthesis, creating mutually beneficial nutrient exchange networks that will become essential components of virtually all terrestrial ecosystems. Arthropod colonization follows plant establishment as terrestrial food webs begin developing sufficient complexity to support consumer organisms. Early terrestrial arthropods face similar challenges to plants in terms of water balance, structural support, and reproductive strategies. But their pre-existing exoskeletons and jointed appendages prove remarkably well suited for terrestrial locomotion. The evolution of tracheae for direct air breathing allows arthropods to exploit atmospheric oxygen more efficiently than aquatic respiratory systems permit. The mystery of terrestrial timing continues to intrigue researchers studying why life remained exclusively aquatic for billions of years before rapidly colonizing land surfaces during the Paleozoic era. Whether terrestrial colonization required specific atmospheric oxygen concentrations, the evolution of particular symbiotic associations, or the development of critical biochemical innovations remains actively debated. The apparent synchrony of plant and arthropod terrestrial appearances suggests that biological interactions may have been crucial for successful land colonization. Perhaps the most profound consequence of terrestrial colonization lies in its transformation of planetary biogeochemical cycles that had operated exclusively through oceanic and atmospheric processes for billions of years. The establishment of terrestrial ecosystems creates new pathways for carbon, nitrogen, and phosphorus cycling while fundamentally altering patterns of weathering, erosion, and sedimentation. Land plants begin the process of atmospheric carbon dioxide regulation that will eventually stabilize global climate systems while creating the oxygen-rich atmosphere that supports complex animal life. The colonization of land represents not just the expansion of life into new habitats, but the beginning of biological control over planetary environmental systems that continues to operate today. Forests rise from the patient accumulation of innovations in plant architecture, their towering forms reshaping terrestrial landscapes that have known only low-growing pioneer communities since life first ventured onto land. Vast swamps stretch across the continents like green seas, their canopies reaching heights that dwarf the simple bryophytes and early vascular plants that preceded them. Tree ferns unfurl fronds the size of umbrellas while maintaining the reproductive strategies of their smaller ancestors. Their spores carried on winds that now must navigate through complex three-dimensional forest structures rather than across open landscapes. These ancient woodlands create Earth's first terrestrial ecosystems, complex enough to support diverse animal communities, while simultaneously beginning the geological processes that will eventually provide humanity with its fossil fuel inheritance. Lycopsid trees dominate the Carboniferous landscape with architectural solutions that seem almost alien by modern standards. Lepidodendron grows to heights exceeding 30 meters while maintaining a trunk structure fundamentally different from any modern tree. Its bark composed of diamond-shaped leaf scars that spiral around the stem in mathematical patterns that optimize structural strength. Unlike modern trees that grow by adding annual rings of wood, lycopsids increase their girth through secondary tissue that maintains juvenile characteristics throughout the tree's lifetime, creating giants that remain essentially enlarged versions of their herbaceous relatives. Scale trees develop hollow trunks that minimize weight while providing structural support adequate for canopy heights that place photosynthetic tissues in optimal light conditions. This architectural innovation allows rapid vertical growth in competitive forest environments while reducing the energetic costs of maintaining massive woody stems. The hollow construction proves so successful that entire forests of these peculiar trees spread across the warm, humid lowlands of Pangaea's equatorial regions. Their unusual anatomy preserved in coal deposits that record the carbon storage capabilities of ancient ecosystems. Seed evolution represents one of the most significant innovations in plant reproductive biology, freeing terrestrial reproduction from dependence on external water for fertilization while providing developing embryos with portable nutrient supplies that support growth until photosynthesis becomes possible. Early seeds develop from modified spore-producing structures, their protective coats preventing desiccation, while their stored food reserves enable germination in locations far from parent plants. This reproductive independence allows seed plants to colonize drier environments that remain inaccessible to spore-dependent plants. Progymnosperms bridge the evolutionary gap between spore-producing plants and true seed plants, their anatomy combining advanced vascular tissue organization with reproductive strategies that foreshadow later seed plant innovations. Archaeopteris develops secondary wood production that creates tree-sized forms capable of competing effectively with contemporary lycopsids and ferns, while experimenting with reproductive structures that approach true seed organization. These transitional plants demonstrate that major evolutionary innovations often emerge through gradual modification of existing structures rather than sudden revolutionary changes. Carboniferous climate creates greenhouse conditions that support plant growth rates and biomass accumulation unprecedented in Earth's terrestrial history. Atmospheric carbon dioxide concentrations reach levels that enhance photosynthetic efficiency, while global temperatures remain warm enough to support year-round, growing seasons across vast continental areas. The absence of polar ice caps allows moisture-laden air masses to penetrate deep into continental interiors, creating humid conditions that support extensive swamp forests in regions that would be deserts under modern climate regimes. Coal formation begins as organic matter accumulates faster than decomposition processes can eliminate it, creating the vast carbon deposits that will eventually fuel human industrial civilization. The warm, waterlogged conditions of Carboniferous swamps prevent complete decomposition of plant material while promoting chemical processes that gradually transform organic tissues into increasingly carbon-rich deposits. Peat formation represents the initial stage of this process. While subsequent burial and geological pressure convert accumulated plant matter into the coal seams that record ancient forest productivity, only took plants roughly 50 million years to transform from simple ground-hugging pioneers into complex forest ecosystems capable of sequestering atmospheric carbon on geological scales. Only required the evolution of secondary growth, seed reproduction, and architectural innovations that allowed competition for light in three-dimensional canopy environments. only needed the development of root systems capable of accessing groundwater while providing mechanical anchorage for tree-sized plant bodies. Arthropod gigantism reaches extraordinary proportions as Carboniferous forests provide habitat complexity that supports the largest terrestrial arthropods in Earth's history. Meganeura achieves wingspans approaching 75 centimeters. Its dragonfly-like form powered by flight muscles that benefit from the elevated oxygen concentrations characteristic of this period. Arthropleura grows to lengths exceeding 2 meters. Its millipede-like body adapted for processing the vast quantities of decaying plant matter that accumulate on Carboniferous forest floors. Amphibian radiation produces the first large terrestrial vertebrates as early tetrapods exploit the abundant resources of swamp forest ecosystems. These semi-aquatic pioneers retain many aquatic adaptations while developing increasingly sophisticated mechanisms for terrestrial locomotion and respiration. Eriops reaches lengths exceeding 3 meters. Its massive skull and powerful limbs adapted for hunting in both aquatic and terrestrial environments, while its permeable skin requires proximity to water sources for successful reproduction. Insect flight reaches unprecedented sophistication as winged arthropods diversify into ecological niches created by complex forest canopy structures. The evolution of wing folding mechanisms allows insects to exploit crevices and confined spaces unavailable to fixed-wing forms, while powered flight enables rapid movement between widely scattered resources in three-dimensional forest environments. Early insects develop life cycles that take advantage of both aquatic and terrestrial habitats, their larvae developing in forest pools while adults exploit aerial niches. Terrestrial food webs achieve complexity that approaches modern ecosystem organization as plants. Herbivores and predators develop increasingly sophisticated ecological interactions. Decomposer organisms evolve specialized capabilities for processing the chemically complex tissues produced by woody plants, creating nutrient cycling systems that support sustained forest productivity. The emergence of predatory arthropods and early vertebrates introduces top-down regulation that influences plant community structure through cascading ecological effects. Atmospheric oxygen reaches concentrations that exceed modern levels by substantial margins, creating chemical conditions that support physiological processes impossible under current atmospheric composition. The elevated oxygen availability enables larger body sizes in arthropods while supporting more efficient metabolism in early vertebrates. These atmospheric conditions result from the carbon burial occurring in Carboniferous swamps, which removes carbon dioxide from the atmosphere while preventing the decomposition processes that would otherwise consume oxygen. Root system evolution allows plants to access groundwater resources while providing mechanical support adequate for tree-sized growth forms. Mycorrhizal associations become increasingly sophisticated as fungal partners develop specialized structures for nutrient exchange with plant hosts. These underground networks create communication and resource-sharing systems that integrate individual plants into forest-scale superorganisms capable of coordinated responses to environmental changes. Fire begins playing ecological roles as terrestrial vegetation accumulates sufficient biomass to support combustion processes that periodically reset forest succession cycles. Lightning-ignited fires create landscape mosaics of different aged forest stands while selecting for plant adaptations that enhance survival and recovery after burning. The evolution of fire-adapted reproductive strategies and growth forms demonstrates that disturbance becomes an integral component of terrestrial ecosystem dynamics. The mystery of late Carboniferous climate change continues to puzzle researchers studying the transition from greenhouse to icehouse conditions that eventually brings an end to the extensive swamp forests of this period. Whether climate cooling results from carbon sequestration by the forests themselves, changes in ocean circulation patterns, or astronomical forcing mechanisms remains actively debated. The decline of Carboniferous forest ecosystems marks the end of Earth's most extensive experiment with terrestrial carbon storage. Perhaps the most lasting legacy of Carboniferous forests lies in their demonstration that terrestrial ecosystems can achieve productivity levels that rival those of marine environments while creating landscape-scale carbon storage systems that influence global climate. The coal deposits formed during this period represent one of the largest transfers of atmospheric carbon to geological storage in Earth's history. Their eventual human exploitation, reversing the climate effects that these ancient forests created, the relationship between forest productivity, carbon cycling, and climate regulation that emerged during the Carboniferous continues to influence Earth system dynamics, providing both historical precedent and future guidance for understanding how terrestrial ecosystems respond to changing atmospheric composition. The Great Dying unfolds across the boundary between Permian and Triassic time, like a slow apocalypse that tests every adaptation life has accumulated over hundreds of millions of years of evolutionary innovation. 96% of marine species vanish from the fossil record during this ultimate biological crisis. Their disappearance so complete that the ocean ecosystems emerging from the extinction event resemble alien worlds more than continuations of Paleozoic marine communities. On land, vertebrate extinction rates approach 70% as forests collapse and terrestrial food webs unravel across continental scales. The Permian extinction represents evolution's most severe editing event, eliminating entire categories of organisms while creating ecological vacancies that will be filled by fundamentally different life forms. Volcanic catastrophe on an unprecedented scale triggers the extinction through the eruption of the Siberian traps. Basaltic floods that cover an area larger than modern Europe, with lava flows that continue erupting for hundreds of thousands of years. These massive eruptions inject enormous quantities of carbon dioxide, sulfur dioxide, and other greenhouse gases into the atmosphere, while simultaneously releasing toxic metals and acids that poison both terrestrial and marine environments. The volcanic winter created by ash and aerosols gives way to extreme greenhouse warming as carbon dioxide concentrations reach levels not seen since the earliest Archean era. Ocean acidification reaches levels that dissolve the shells of most marine organisms as atmospheric carbon dioxide dissolves into seawater and forms carbonic acid. The resulting pH changes make it impossible for most calcifying organisms to maintain their protective shells and skeletal structures, leading to the collapse of reef ecosystems and the extinction of most marine invertebrate groups. Brachiopods, which have dominated marine filter-feeding niches since the Cambrian, suffer catastrophic losses that will relegate them to minor ecological roles throughout subsequent geological history. Anoxic ocean conditions develop as marine productivity collapses, and decomposing organic matter consumes available oxygen in seawater. Purple and green sulfur bacteria proliferate in the oxygen-depleted depths, their metabolic activities producing hydrogen sulfide that bubbles to the surface and creates toxic atmospheric conditions. The resulting marine chemistry resembles that of the early Proterozoic oceans, essentially reversing billions of years of progressive oxygenation and returning Earth's oceans to chemically primitive conditions. Terrestrial ecosystem collapse proceeds through cascading failures that begin with plant communities and propagate upward through herbivore and predator populations. The combination of extreme temperatures, altered precipitation patterns, and soil chemistry changes eliminates most forest ecosystems while replacing them with sparse, drought-adapted plant communities that cannot support the diverse animal assemblages of the late Permian. Therapsids, the dominant terrestrial vertebrates of the Permian, suffer massive extinctions that eliminate most lineages and reduce survivors to small, generalized forms. Recovery patterns following the extinction reveal the resilience of certain biological designs while demonstrating the vulnerability of others to extreme environmental stress. Disaster taxa. Organisms adapted for rapid reproduction and colonization of disturbed environments dominate early Triassic ecosystems, while more specialized forms slowly re-establish themselves over millions of years. The recovery process requires roughly 10 million years to restore ecosystem complexity comparable to pre-extinction levels, demonstrating that evolutionary repairs proceed much more slowly than evolutionary destruction. Only took Earth's most severe environmental crisis to eliminate the accumulated biological complexity of the entire Paleozoic era in a geological instant. Only required a few 100,000 years of volcanic activity to reverse hundreds of millions of years of evolutionary innovation and ecological sophistication. Only needed the convergence of multiple environmental catastrophes to demonstrate that even the most successful biological designs possess vulnerability thresholds beyond which survival becomes impossible. Archosaur radiation begins during the Triassic recovery, as reptilian survivors explore ecological niches vacated by extinct therapsids and other Permian vertebrates. Early archosaurs develop increasingly sophisticated physiological and behavioral adaptations that allow them to exploit both terrestrial and aquatic environments while competing successfully against surviving therapsid lineages. This radiation sets the stage for dinosaur dominance that will characterize Mesozoic terrestrial ecosystems while also producing the crocodilian lineages that persist to modern times. Marine reptile evolution accelerates as recovering ocean ecosystems provide opportunities for vertebrates to exploit marine niches previously dominated by invertebrate predators. Ichthyosaurs develop dolphin-like body forms adapted for pursuit predation in open ocean environments, while plesiosaurs evolve longer necks and specialized feeding strategies for nearshore marine habitats. These marine reptile radiations demonstrate that vertebrates can rapidly evolve sophisticated aquatic adaptations when ecological opportunities become available. Plant community reorganization follows the extinction as gymnosperm groups that survived the crisis begin experimenting with new architectural and reproductive strategies. Conifers develop increasingly sophisticated water conservation mechanisms that allow them to dominate drier, post-extinction landscapes. While cycads and other seed plant groups explore alternative solutions to the challenges of terrestrial reproduction, the plant communities that emerge from the extinction create the ecological foundation for Mesozoic terrestrial ecosystems. Mammalian origins trace back to small therapsid survivors that persist through the extinction crisis by adopting nocturnal, insectivorous lifestyles that require minimal resources while avoiding competition with larger archosaur predators. These early mammals develop sophisticated thermoregulation, improved sensory systems, and reproductive strategies that provide them with ecological flexibility despite their small size. The mammalian lineage that emerges from the Permian extinction remains ecologically marginal throughout the Mesozoic, but retains evolutionary potential that will eventually allow explosive radiation following later extinction events. Reef ecosystem reconstruction requires millions of years as surviving marine organisms gradually redevelop the complex ecological interactions that support high-diversity carbonate platform communities. Scleractinian corals emerge as the dominant reef builders, replacing the rugose and tabulate corals that dominated Paleozoic reefs. The new reef ecosystems develop different architectural patterns and ecological relationships compared to their Paleozoic predecessors, demonstrating that evolutionary replacement often involves fundamental reorganization rather than simple substitution. Climate system recovery involves complex interactions between atmospheric composition, ocean circulation, and biological productivity that gradually restore Earth's environmental systems to more stable configurations. The extreme greenhouse conditions of the early Triassic slowly moderate as volcanic activity decreases and biological carbon sequestration processes re-establish themselves. However, the climate system that emerges from the crisis operates under different boundary conditions than those that characterized the late Permian, creating environmental contexts that favor different biological solutions. Ecological complexity rebuilding follows predictable patterns as simple ecosystems gradually develop the trophic relationships and species interactions that characterize mature biological communities. Primary producers re-establish themselves first, followed by herbivore populations that support increasingly sophisticated predator communities. The rebuilding process demonstrates that ecological complexity emerges through stepwise addition of functional groups rather than simultaneous development of all ecosystem components. The mystery of survival selectivity continues to intrigue researchers studying which biological characteristics enabled certain organisms to persist through the extinction while closely related forms perished. Body size, metabolic rate, geographic distribution, and ecological specialization all appear to influence survival probability. But no single factor provides reliable prediction of extinction versus survival. The apparently random nature of many extinctions suggests that chance events become increasingly important during severe environmental crises. Perhaps the most profound lesson of the Permian extinction lies in its demonstration that biological complexity represents an accumulated treasure that can be lost far more rapidly than it can be gained. The hundreds of millions of years required to build Paleozoic ecosystem sophistication vanish in a geological instant, while recovery requires additional millions of years of evolutionary innovation and ecological experimentation. This asymmetry between destruction and construction characterizes all major extinction events, reminding us that the biological complexity we observe today represents the cumulative product of countless evolutionary innovations that survived numerous environmental crises throughout Earth's history. The organisms that inherit the post-extinction world do so not necessarily because they represent superior biological designs, but because they possess the combinations of characteristics and circumstances that allow persistence through environmental catastrophes that exceed the adaptive capabilities of even the most successful evolutionary lineages. Deep time settles like sediment across the fossil record, each layer preserving the whispered stories of organisms that inhabited Earth billions of years before consciousness emerged to contemplate its own origins. The vast chronology of pre-human life unfolds in geological strata that stretch backward through eons, measured not in human generations, but in the patient accumulation of evolutionary innovations. Each breakthrough requiring millions of years of experimentation before the next tentative step toward complexity could begin. From the first organic molecules that learned to replicate themselves in primordial seas to the diverse ecosystems that survived the Great Dying. Life's early chapters record a narrative of persistence that spans timescales beyond human comprehension. The silence of these ancient worlds echoes through museum halls where trilobite exoskeletons rest in glass cases, their compound eyes no longer scanning seafloors that vanished before the first vertebrate drew breath on land. Stromatolites preserve the architectural legacy of cyanobacterial communities that transformed Earth's atmosphere through billions of years of patient photosynthesis, their layered structures standing as monuments to the microscopic organisms that made complex life possible. Each fossil represents not just an individual organism, but an entire evolutionary lineage that succeeded for geological ages before yielding to environmental changes or competition from more adaptable forms. The prokaryotic multitudes that still inhabit every environment on Earth carry within their cellular machinery the molecular memories of life's earliest experiments with metabolism, reproduction, and environmental sensing. These invisible inheritors of the pre-human world continue the biochemical processes that their ancestors pioneered billions of years ago. Their genetic codes preserving evolutionary solutions that proved so successful they require no fundamental revision. In the depths of hydrothermal vents and the frozen soils of polar regions, archaea and bacteria maintain the same basic cellular architectures that dominated Earth's biosphere when the planet was young and every ecological niche remained unexplored. The eukaryotic revolution that created cells complex enough to build multicellular bodies continues operating within every plant and animal alive today. The ancient endosymbiotic partnerships between formerly independent organisms, now so thoroughly integrated that their separate evolutionary origins become visible only through careful molecular analysis. Mitochondria and chloroplasts retain their own circular DNA molecules, like genetic souvenirs from the age when they existed as free-living bacteria. Their presence in modern cells serving as living fossils that preserve evidence of cooperation's triumph over competition in evolution's deepest experiments with cellular organization. Time flows onward like the patient currents that once carried Ediacaran organisms across seafloors carpeted with microbial mats. The strange, quilted creatures that explored multicellular possibilities that evolution never repeated. Their peculiar geometries rest now in the geological record like pressed flowers from gardens that bloomed and faded before predation was invented. Their peaceful communities representing a brief experiment with large-scale life that required no teeth, claws, or shells for success. The transition from their soft-bodied world to the hard-shelled communities of the Cambrian marks one of evolution's most dramatic editorial decisions. The replacement of one entire approach to multicellular life with architectural principles that continue shaping biological design today. The rhythm of deep time pulses through rock formations that record the rise and fall of entire biological dynasties. Each extinction event clearing ecological space for innovations that would have been impossible while earlier communities maintained their dominance. Trilobites yielded to vertebrates, ancient forests to modern plant communities, marine reptiles to mammals, in cycles of replacement that demonstrate evolution's capacity for fundamental reorganization when environmental conditions exceed the adaptive limits of existing life forms. The pattern repeats across geological time like a planetary heartbeat that measures progress not in years or decades, but in the millions of years required for major biological innovations to emerge and prove their worth. Stillness descends over these ancient narratives as the long day of pre-human evolution settles into the geological record, each chapter closed by layers of sediment that preserve its characters for future discovery. The molecules that danced together in RNA's early experiments with self-replication now rest quietly in the chemical bonds that hold modern genetic codes together. Their ancient innovations so thoroughly integrated into life's basic operations that their revolutionary nature becomes visible only through comparison with the sterile chemistry that preceded them. The first cells that learned to cooperate in multicellular ventures sleep now in fossil impressions that capture their experiments with tissues, organs, and body plans that set the templates for all subsequent biological architecture. The long conversation between life and environment that shaped Earth's transformation from a sterile planet to a living world continues in the background of everyday existence. The cycles of carbon, nitrogen, and phosphorus still following pathways established by organisms that vanished hundreds of millions of years ago. Their metabolic innovations created the atmospheric and oceanic chemistry that modern life depends upon. The oxygen-rich air and nutrient cycling systems that make complex ecosystems possible, representing the accumulated legacy of countless extinct lineages that contributed their molecular innovations to the common biological treasury. Sleep comes finally to this ancient chronicle as the weight of geological time presses down upon the fossil record, preserving in stone the evidence that life's creativity extends far beyond any single epoch or evolutionary experiment. The pre-human inhabitants of Earth rest now in the patient embrace of sedimentary rocks. Their stories told in the universal language of chemistry and physics that connects the simplest bacterial cell to the most complex multicellular organism. In their sleeping, they continue to inhabit the planet they helped create. Their molecular innovations woven so thoroughly into the fabric of modern life that their presence remains essential even in their absence. The breathing of deep time slows to match the pace of geological processes that measure change in millions of years rather than individual lifetimes. Each breath spanning entire evolutionary radiations that explore new possibilities before settling into the fossil record like dreams made manifest in stone.